네, 여러분 안녕하십니까? 잠시 안내 말씀드리겠습니다.
May I have your attention, please?
Esteemed speakers, distinguished guests, and ladies and gentlemen, very good morning and welcome. This is the fourth Infectious Disease Research Institute cooperation. Very good morning and once again, welcome everyone. It is my another great pleasure to serve as your host today. My name is Ki Yoon-sung. Very happy to see you all. 여러분 안녕하십니까? 오늘 사회 행사에 와주셔서 대단히 반갑고 감사합니다. 인사드리겠습니다. 저는 진행을 맡은 Thank you very much. Well, it is another full house today. Thank you very much for your attendance. Your participation actually elevates the meaning and significance of this event. So once again, thank you for taking your precious time to be here with us today. Well, this is a two-day event from yesterday until today. It is co-hosted by National Medical Center and Korea National Institute of Infectious Disease, and it is happening in the conjunction of the second IGONI International Symposium for Infectious Disease Research Institute, which happened yesterday right here. Well, the organizers, I heard that, put extra efforts to choose the topic for this event. And finally, they come up with a special topic of the the strategies for developing AI-based rapid technology and the response technologies for infectious disease. I believe it is very timely since we are living in the AI era where we can't talk about anything without AI. So under this overarching topic, we're gonna delve into three thematic sessions. So please have a great expectation for today's event as well. Without further ado, I officially declare the beginning of fourth IDRIC with all big hands from all of you. 여러분, 그러면 이제 여러분들의 큰 박수로 제 사회 행사
We will open the fourth IDRIC symposium with a big round of applause.
So please allow me to briefly walk you through today's event. We're gonna begin with the three speeches from three figures, and then we take a short break to take up photos and some stretchy legs, and then we will move on to the main session. Throughout today, there are gonna be three main sessions, which cover a total of 12 presentations. And after each presentation, we're gonna give some time to ask questions and express your comments. So we cordially ask all participants, whether you join us offline or online, to ask a lot of questions and discuss a lot. And also, just like yesterday, we separate two separate rooms for your business meeting. So if you are willing to take part in that meeting, then please move to the studio 2 and 3. And please keep track of your time if you are designated speakers. And bear that in mind, let me just move on to the speeches. First up, opening remarks. And it is my great honor to invite Dr. Young Ki-jung, Director General of the Korea National Institute of Infectious Disease. Please welcome him with a big hand. 네, 정영기 소장님께서 개회 말씀해 주시겠습니다. Well, before taking his current position, he studied pharmacy from Kyunghee University and an MBA from KAIST. He has held several key leadership roles within the KDCA before taking the current position.
Good morning, distinguished guests. I am president of the National Institute of Infectious Diseases. First of all, let me sincerely thank Mr. Jae-won Nam, the president of National Institute of Health, Mr. Hong-bin Kim, the chairperson of Korea Society of Infectious Diseases, and the speakers for their presentations and chairs, and all distinguished guests for being here with us. Now, in its fourth year, the International Symposium on Infectious Disease Research Institute cooperation has been working very hard to strengthen the national and international cooperation by establishing a cooperative framework among domestic and international infectious disease research institutes, especially for the past 3 years. The symposium has evolved into a leading international symposium in the field of infectious diseases sharing the latest research findings and promoting various joint research projects through close cooperation with global institutes such as US NIAID, Japan's NIID, AMID, Denmark's SST, Singapore's NCID, WHO, and CEPI. Infectious disease is not just a problem for a single country. We all face this problem. We need a close cooperation and a coordinated response among nations. To this need, we have to pool our national resources and establish international cooperation system. Now, there are the missiles. There are countries where missiles are prevalent, and we have over 70 cases of H5N1 bird influenza. We also are faced with emerging infectious diseases such as monkeypox, Marburg virus, dengue fever, and Nipah virus as well due to climate change. Consequently, the international community is actively adopting innovative infectious disease response strategies that leverage artificial intelligence and has already achieved remarkable results. Prominent examples such as AlphaFold protein structure prediction, acceleration of mRNA vaccine design, and AI-based antibiotic and vaccine candidates discovery are good examples. AI technology is increasing the speed and accuracy of infectious disease response as a key element in preparing for future pandemics. So, this symposium has the theme of strategies for developing AI based rapid response technologies for infectious diseases. We will share world class research achievements in the field of antiviral therapeutics, bacterial infection response, and rapid vaccine development platforms, and for exploring practical ways for international cooperation. I hope that this event will serve as a strong starting point for domestic and global research institutes to further strengthen their cooperation and build a safer society. Thank you very much again for your attendance. I wish all of you a successful symposium and good health and prosperity. Thank you very much.
Dr. Zhang, he expressed the sincere appreciation of your participation and also raised the bar for this event. Thank you. And moving on, I'd like to invite the next speaker who's going to deliver the welcoming speech, Dr. Jae-hwan Nam, Director General of Korea National Institute of Health. So, ladies and gentlemen, please join me in welcoming him with a big hand. Immunology and currently he's serving as a Director General of Korea National Institute of Health, and before that he teaches at the biomedicine at Catholic University of Korea.
Hello, good morning. It's a very nice to meet you. I'm Nam Jae-hwan, Director General of Korea National Institute of Health. Following yesterday's second Lee Gun-hee International Symposium on Infectious Disease Research, I'd like to extend my sincere congratulations on the joint convening of the 4th International Symposium of Infectious Disease Research Institute. It is great honor and pleasure to deliver welcoming remarks. I'd like to express my sincere appreciation to all speakers who have prepared their presentation and session chairs, and the chairperson Kim Hong-bin of Korea Society of Infectious Disease for delivering word of congratulations, and Director Chang Young-gi on the National Institute of Infectious Disease and all those involved in organizing this event, as well as all the distinguished participants. Distinguished guests, through the global COVID-19 pandemic, we have come to realize the threat of infectious disease pandemic extends far beyond public health, posing serious risk to social stability, even national security. Today, we are confronted with multiple concurrent threats, including continued outbreak of highly pathogenic avian influenza and global spread of mpox and Nipah virus, and the emergence of novel infectious disease driven by climate changes. In response to these complex challenges, the rapid development of vaccines and therapeutics through artificial intelligence is no longer option. At the Korea National Institute of Health, we are actively advancing AI-driven research across multiple domains, and also design for the mRNA and protein-based vaccine is undertaken. In addition to actively respond to Nipah virus infection, high-risk zoonotic disease, we are systematically promoting vaccine development by leveraging domestic technological capabilities and mRNA technology and artificial intelligence. In this regard, the AI-based strategy for rapid response to infectious disease to be discussed at this symposium are both timely and highly significant. Korea National Institute of Health will continue to strengthen the linkage between research and implementation system, ensuring these outcomes are swiftly translated into policy and practice. I sincerely hope that today's symposium will serve as a foundation for rapid development of a vaccine and therapeutics. And once again, I'd like to extend my sincere thanks to every one of you for your participation. I wish you good health and happiness and ask your continuous support. Thank you.
Thank you, Dr. Nam. He just highlighted the initiative he's having at his institute and also expressed a high expectation for today's program. Thank you. And moving on, I'd like to invite Dr. Hong Bin Kim to the stage for his congratulatory speech. And he's the chairman of the Korean Society of Infectious Disease. Please welcome him with a big hand. He's a triple degree holder from Seoul National University and worldly renowned expert in infectious disease. Currently, he's playing a pivotal figure in global health and antimicrobial stewardship.
네, 안녕하십니까. 대한감염학회 이사장.
Good morning, distinguished guests. I am Kim Hong-bin. I would like to congratulate the opening of the fourth IDRIC symposium, and I am very pleased to be here with you. This is such a meaningful occasion. Infectious disease is caused by microbiomes, and the infectious diseases pose a lot of challenges to our society. As we experience through COVID-19 pandemic, infectious disease is not just a matter of a single academic discourse of infectious disease study, but it requires a close cooperation among various stakeholders engaging in basic research, clinical research, public health policies, and international cooperation. This international symposium today will respond to the needs of the times to strengthen the cooperation among research institutes of infectious diseases. It will also expand the international networks. This is such a meaningful academic platform. I participated in HIMSS conference. At that conference, AI was really important topic. In the symposium, we will discuss AI-based rapid response to infectious diseases along with future directions for our research activities. So this is an important symposium where we will seek a new paradigm in responding to infectious diseases. Especially AI-based antiviral agents, vaccine, antigen optimization, and establishment of surveillance and prediction model require us to meet the demands of clinical practices by developing research technologies and by developing research methods as well. And this discussion needs to lead to the actual patient treatment and public health policies, and then we will be able to establish a more effective and precise infectious disease response system. The Korea Society of Infectious Disease has so far been committed to developing clinical guidelines for infectious diseases, activating academic research while nurturing professionals. I am confident that our Korea Society of Infectious Diseases will continue to closely cooperate with research institutes and governments to reflect our academic achievements in the clinical field and policies. I wish that through the symposium today, we will seek out specific cooperation and implementation strategies. I wish all of you insightful symposium. I wish all of you a successful international symposium. Thank you very much.
He also pointed out how timely and proper our topic is. Thank you for mentioning that. Well, we are deeply delighted to listen from three well-known figures in this field, but we have so many other leaders and experts in the field as well. However, to the interest of time, we can introduce all of you individually. Please forgive us generously. Well, if you were here yesterday, you may understand how intensive this program is. So you're going to have another intensive but informative day. So before jumping onto our main session, why don't we take a 15-minute break for refresh? So you can just take a short break to have some coffee and chat. And we will begin our main session in 15 minutes at 10 a.m. sharp. So let's take a 15. 네, 여러분 본격적인 세션에 들어가기 전에 15분 쉬었다 오도록 하겠습니다. 잠시 쉬었다 10시에 뵙도록 하겠습니다. 감사합니다. 네, 잠깐 쉬시기 전에 오늘 귀빈 여러분들 모시고 기념사진
We would like to take a commemorative photo with our distinguished guests, speakers, chairs, and please come to the stage.
to have a photo session. I'd like to invite all moderators, speakers, and VIPs to the podium to take a picture. So please join us to the podium to take a picture together. 네, 약속한 10시가 거의 다 되어서요. 아직 행사장
So we are going to begin the first session. Those who are outside, please do come in and take a seat.
If you are still outside of the event hall, please hurry yourself to take your seats. As soon as this event hall is arranged, we're going to begin our main session 1. 네, 지금 다 들어오고
So now we have the participants coming into the hall.
So welcome back, everyone. This is the 4th Infectious Disease Research Institute Cooperation and the thematic session 1. And as I mentioned in the opening ceremony, we're going to have 3 sessions, and each session will cover 4 presentations. And the session 1 is about the strategies for developing AI-based rapid response. Actually, that is the overarching theme which, for our main event, which is the strategies for developing AI-based rapid response technologies for infectious disease. Under this big topic, we're going to delve into the first topic of next generation R&D strategies for AI-based antiviral drug development. To lead this meaningful session, 2 co-chairs are ready on the stage. Let me just introduce them one by one. First up, Dr. Juyeon Lee. She is currently serving as the director of the Korea National Institute of Infectious Disease, and she holds a doctoral degree in life sciences from Korea University, and she is well known as a premier expert in infectious disease. And in the meantime, Professor Man Sung-bak, who will serve as a co-chair, and he also holds a PhD in microbiology from Korea University, and he's also leading expert in his field and currently teaching at his alma mater. And actually, I found one interesting thing. Both of them have a lot in common because they graduate the same school here in Korea, Korea University, and when they went on to the United States, they also working as an instructor and a researcher at same school of Icahn School of Medicine at Mount Sinai in the United States. Even though there is a little bit of difference of timing, so I think they can bring the world level of knowledge and insight for this session. So ladies and gentlemen, please welcome them a big hand. 여러분 자장님들께 큰 박수 부탁드리겠습니다. Over to you.
네,
So now we are going to begin the first session. So in this first session, we are going to have a discussions about next generation R&D strategies for AI-driven antiviral drug developments. We have 4 presentations. I'm going to cover 2 presentations, and the remaining 2 presentations will be supported by Professor Park Man-sung. First speaker is Professor Kim Yong-ho. So let me introduce him. So Professor Kim studied in the Sungkyunkwan University and the postco in order to continue his study. And he actually continued his study in biochemistry at the University of Pennsylvania. So he studied at Sungkyunkwan University and KAIST, and after to the University of Pennsylvania. So let's invite him to hear presentation. Good morning, ladies and gentlemen. I'm from Sungkyunkwan University. First of all, I'd like to extend my sincere gratitude to organizers for inviting me in this meaningful occasion. Today, what I want to share is AI. So how the infectious disease they can be supported by AI. So AI-enabled generative technology will be shared in my presentation. So actually, we pretty much focus on generative protein design. So by utilizing generative AI, and we are focusing on developing neutralized the protein and the therapeutics. So that is the main focus of my presentation. So in the past, actually, we screened a small molecule. So 1 in a million samples has been searched. So the traditional drug discovery has relied on search and discovery approaches. So researchers screen a vast amount of candidates to find proteins and antibodies with the desired functions. But in the era of AI, we moved to design and generative approaches. By utilizing the generative AI, we are developing backbones, and the deep learning algorithm can be utilized in order to design the sequence. So we actually search the sequence for the proteins, so we can have a create the benefits based on generative AI. So we have a short history of utilizing generative AI, but that is the advancing in very fast speed. So this can be compared to the central dogma. So here you can see the structures and the sequence. The protein begins with the amino acid sequence. So you can see here the three-dimensional structure, and based on that, the function can be secured. For the last few decades, we have created questions about sequence, structure, and the functions. So special, the sequence can read to structures, and also it is important to have the minimum level, the structure, in order to have desired functions. So the scientists actually want to secure the sequence information and structure information. So based on the functions, and we can categorize proteins. Though we are not able to see the dynamics, but we have a certain knowledge and information about enzyme. And based on the information about sequence and structure, and we can have a more detailed the approaches, especially when it is backed by AI, and we can have additional benefits. So in my field, there is a sequence and a structure, and based on the information about the sequence and the structure, we can have the additional the progress. So when we develop new and novel structure which does not exist in nature, and we can actually go for the new field which is the protein design. So that they initiated about 25 years ago, but there has been limitation and a lack of information and knowledges. Because of that, that technology could not take off. But based on generative AI in very fast speed, we can go for the protein and the design. So, well, this is the X-ray crystallography. So based on that, we can have a better understanding. So we have a massive amount of the database. In the year 2025, the Professor Huan actually provided a great insight, and I had a great opportunity to have a discussion with him. So this structure is vast indeed. So it is quite difficult to have the entire understanding about this structure. Based on the AI technology, we can go into the era of the fusion. So design-driven paradigm can be the wide open now. So if we have an understanding about the target, then we can have the better the approaches regarding the functions and the folding. So we have an increasing understanding about that one. So we can have the modulation for the targetable region on viral protein. So we can go into the era of protein design. So we want to develop neutralizing the viral, the protein. So I'm going to give some information about that, even though we are in the initial stage. So here you can see the recording and the pandemics. So here you can see the area of my expertise. I studied pseudo the virus, and here you can see the recurring pandemics. So as you can see, the interval between pandemic appears to be shortening in recent years. That's why our discussion related to initiatives such as 100 days missions. So when we see the protein and the behaviors and the immune escape is witnessed, so it is quite difficult to provide the proper therapeutics. So by utilizing AI, we can move faster so that we can deal with these the mutations. So cryotomography technology can be utilized in order to have a better understanding. So this one is a spike protein. So you can see surface area of a virus. So, I clicked a wrong button. So sorry for that. So please show this slide again. So here you can see the surface area, the lipoprotein density can be seen here. So the tomography can be seen by utilizing the advanced technologies. So by taking snaps, we can see the surface of the virus. And here you can see the generative protein design and the foundations. These surface protein are preliminary and the primary target of the immune response. So here there are many different kind of proteins, and I have influenza. I have a great interest about the influenza virus, but in the process of drug development, there have been the mutation. So recently, I focus on the invasions. So that occurs at the embracing. So I initiated my study about this. I received the data set, so you can see the rendering is done. If you take a closer look, and you can find this protein is not static. So the protein is a dynamic. So the protein and actually shows the dynamic motion, so that goes into the target site. And as for the COVID S2 receptor is on the targeted. Within a few seconds and within nanosecond, the folding can be transformed. So the closed state and open state, and you can see that. So once that is open, that can go into the target site, and that moves in very fast speed. The problem is, for the small site, we can find some solutions, but it is quite challenging to develop and neutralize the protein. If this site is a major site and there is a behavior of immune escape, so it is difficult to have a proper design. So here you can see the mechanism. So they actually shift from open state to the closed state, and once they find the target, this goes into the stabilization of open state and go into the intermediate state. And after that, the flip fusion state is begun, and the cell invasion is done after that. So here you can see the profile of this particular the protein. So AI has been developed in very fast speed, but the basic of AI the algorithm is a proper data. So we need to secure the mutation-related data and the immune-related data. We have accumulated data about that. So those data is publicly available. So here you can see mutation profile. So the red color shows the high frequency for the mutation. But as you can see here, only the specific site experienced mutation, which means that this particular the protein have the mutation in a particular site, and in other the area, the immune escape the behavior is witnessed. So And also they move in continuous manner in order to search for target. So the researchers in the past have gone through this process. Well, please take a look at the middle side. So, well, actually, researchers developed the antibody in order to go for the target site. But at the development stages, it is difficult to find the proper site. So, well, that is based on the trials and errors. The study has continued, and a massive amount of the money and the time are allocated in order to develop one single the solutions. But with the single antibody, it is difficult to go for the proper solution. So we could not find the universal solutions. So we have the following approaches. So antigen is profiled. So that will be explained in the later part of my presentation. So here you can see the novel therapeutic strategy and the generative AI-based protein design. So, well, previously we had a limitation and regarding antibody-based approaches. So based on the data learning is done. So the target site can be studied based on deep learning algorithm. AI algorithm is additionally applied for the binding and inhibition. And so this specialized protein can be designed in that way. So I actually studied 3 different targets and from 6 to 8 different types. Actually recommended. But I only focused on 3 targets instead of 6 to 8 targets. So the SARS virus, is one of them, and H5N1, and well, SARS-CoV-2 and influenza and RSV, those are the major target virus in our research team. So RNA fusion protein is studied in our research team in order to have a protein productions. So here you can see the shape of the protein. So, well, type of antigen protein we target is presented here. So the sequence and the structures are slightly different. So here you can see 2 different the categories. So we have the many different database. Actually, one is the domestic database. So where target exists and where the mutation occurs. So we are developing specific types and the universal types, and after that we go into the broad range of experiments. And if we have to develop antigen based on all these, and it takes a lot of investment. So we have different approaches, and that will be explained in the later part of my presentation. So here you can see the escape and the score. So the variation can be witnessed here. So the deep learning algorithm is used. Well, by securing data, anyone can get a result out of that. So within this profile, you can get the escape score, and also accessibility score is found in this profile. So, inner side of a folding is rarely be the target. So we focus on other areas in order to have a protein design, protein interaction. The profile is required. So we have a great study in order to have information about the protein to protein interaction. Here you can see red side, and when we see the red colored side, and the protein interaction was very active. So that is related to dynamic of the protein, and also the surface of a protein has different characteristics that is related to this high percentages of protein-protein interactions. So very briefly, you can see the mutation information. So the amino acid information can be found here as well. And the survival rate can be witnessed and the mutation rate can be seen as well. And also the exposure rate can be seen. Based on that, we have a mapping data. By learning this mapping data based on the continuous learning and the strain and when new information about the new strain is given, and we can get outcomes which shows the possibility of a mutation. So here you can see the red color area. And as I mentioned, a red color area showed high the probability of mutations. So here you can see in order to have the better survivor, in order to have a better immune escape, the mutation happens. And this one is a hemoglobin. The core area is a blue, so the core the structure should be intact. So the prediction data and profile data matches with the high percentages. This one is a LSB. LSB is a rather unique. So here you can see the mutation profile applied to LSB fusion protein. So you can see dramatic changes of the data. That's why it is difficult to have the precise targeting based on the traditional and conventional antibody. And here you can see the protein-protein interaction profiling. So blue color means and there is a high the success and the possibilities for the targeting. So we need to find the target site. So we are making effort in order to define the final target binding site. So as you can see here, we actually develop profile data and learning based on the data is done. And after that, prediction can be made based on those learning. So in order to design this, it takes about 2 weeks. We utilize the supercomputer. That's why it takes about 2 weeks. Once a mutation happens, then information is given, then computation and the process the initiated, and based on that, for about 2 weeks. And this can find about 10,000 the candidates. So I should decide whether I go for the experiment for each different and the candidates. The backbone is generated and sequence can be found, and the weakness of AI is the data. Data is quite the static. So this data does not have dynamic data. So as for the energy calculation, and the special information should be given, specific information should be given again in order to have the insight out of that. So lead sequence should be found, and its display is utilized to this end. So, the price is about 15,000 and the expensive one is about 50,000. So, let's say we have a 1,000 design, then the total cost is about 10 million. And if we invest about 50 million, we have about 100 sequence. So, and after the antigen and the information is given, then we can have the profile and the outcomes. So, on a single cell level, and we can get our information. We can and have a better sequencing. So in order to do the sequencing, it takes about 10 days. So based on that, we can find the candidates. We use the immunogenicity data file, the solution as well. So immunogenicity, does not happen in some sequence. So we want to find such a sequence in our study. And into final stage is designing the protein. So you can see the such a process. So on a peptide level, and we are designing protein. So on the peptide level, and we can sometimes achieve neutralizations. So as for the designing of a protein, and well, we have different outcomes case by case, and well, more investment and better equipment can lead to better result. So here there are 7 different the targets, and we have open form and closed form and the data. And the open form and the data is available. Actually, I have 2 more minutes, so let me go fast. So this one is about the backbone structure for the target site. So radiance of giants and the protein folding information is given. So even though we utilize AI, if folding is heavy, we cannot get a great result. So it is important to secure the information about folding. So within the confidence level, we need to find the better the folding ranges. So here you can see score 1, 2, 3, but you should not disappoint yet because the algorithm will go on. Of course, it requires additional computing power, but anyhow, this algorithm goes on. So I finally received the sequence information. So let me jump a bit. So this is the result. So transfection is done. So now I have a sequence outcome. So the design defined the protein were expressed on the east surface. So, well, in the year 2024, there was the infection case on the cat, so we utilized those specific strain in order to study. And based on this strain, we developed the protein. So within 1 month, we could find the binding. So, well, it took about 1.5 years in order to develop the algorithm, but by making additional investment and by utilizing additional equipment, we can shorten the time. So the preclinical the center led by the Professor Lee and Professor Kim is making the tremendous effort and in order to continue this test, and the Korea Disease Control and Prevention Agency will provide support as well. So here you can see the outcome, and this outcome became available 2 days ago. Here you can see the neutralization capacity, and the neutralization capacity is a differ. So this one is a peptide. So the opportunity can be calculated, and within relatively short period of time, we can find and neutralize the outcomes. And I'm very excited. I believe you can feel the excitement. So here you can see cryo-EM validation process. So earlier I shared some images, and that is based on deep learning algorithm. So within this protein and the structure and the dynamic structure can be studied. So in the antibody, and we have ability control. So based on that, we can have numerous the experiments. Peptide size itself is small. That's why we can have a small progress, and the profiling is done. So ability control, the optimized control at the state can be found. Based on that, antibody can be produced. It took about 2 weeks in order to produce antibody, so we can move to the next stages. So let me skip. So this one is a mRNA-based therapeutic strategies, and you can see target site here. So I'm going to publish the paper based on this experiment, but we do not provide the source code. So the AI developed this, so it is difficult to see all these the process in white box. So this is a generative AI-based study. So even though there is a mutation based on this profile, we can go into the next stage in very fast speed, and we can have a conversion to the mRNA, and also, we want to integrate these steps into unified therapeutic development process. So that is our goal. Thank you. Well, thank you for your presentation. Actually, you provide explanation about generative protein design. So, the influenza virus, SARS virus, and the RNA virus can be studied. So the sequence-based and structure-based study was shared. So thank you for that. And we are going to open the floor to receive questions and comment. Please introduce yourself first, and after that, raise questions and comment. So I'm Nam Jae-hwan. Well, it is a very intriguing the presentation, and actually, your research direction is in line with the direction of my organization. So, my question is the follow. Actually, epitope is found based on AI, and the paratope is found based on that. So the MLA the therapeutic studied, but antibody and the therapeutic can last longer. So that will be the proper approaches. So what is your opinion about that? That is my first question. So my second question is, you can we can see the east and the surface. So the peptide has been selected. So, is it possible to go into the in silico stage? And if we just jump into the in silico stage, and what is the percentages of the success? So the AI-based design success rate, based on our experience, is less than 5%. And that is great still. So when we actually the enter 1,000, we have 5 the candidate. Those 5 candidates can be potential drug. So we can advance our technologies. So I tried in silico the approaches in the past, but the success rate is less than 0.6%. So AI success rate is pretty high with the 5%. So trials and errors can be corrected in the process. So when we have a final candidate, and just like functions, and success rate can be higher than in silico. So that is based on my experience. And for your first question, as a peptide, we develop peptide because I have the expertise about the peptide. So, as for the antibody, and the unit cost is pretty high. So, peptide can be generated in a very effective manner based on the advanced technologies. And as for the cost and cost-wise, and we have a greater benefits by developing the peptide rather than go for the antibody-based approaches. So just like a GLP-1, we have the various opportunities to generate the protein. So from the perspective of administrator, peptide can provide better advantages. One additional question, you said 5% for the AI. So AI learning can be improved. So if the data is accumulated, do you believe the success rate can improve to the 50%? Yes, there is a possibility. There are the various models, but all of them are in the initial stage. So as the protein the designer, I'm the first generation here in Korea. I'm not the structure experts. So the researchers like me are utilizing this model. So based on collaborations, and we can advance these models in faster speed. But currently, AI-based model is limited. So we need to have great collaborations in order to develop AI-based model. Then we can have a better success rate. So there is a possibility for that. Well, it's a very nice to meet you. I'm Igunu from the New Drug Development at the Institute. So you shared the protein mutation profile. So actually, you show the red color, which has a high percentages of mutation, and I'm structure experts. So the temperature factors and the other the elements and could have seen. So have you ever studied the high motion and the whether high motion lead to high mutation? Yes. Actually. There is a correlations between the two, and I can say for sure. So high the motion lead to high mutations. But about the small molecule accessibility is good, but we are based on biologics. So, as for the folding, and there are some limited accessibility, that's why profiling data is required. We want to target specific area, but about there are different dynamics. So we need to designate the proper site. So peptide and protein has a different approaches because of the limitation regarding space target space. So we have the many questions, but time is limited. Let's accommodate one additional question. I'm H.S. Seung from the KTR. So thank you for the presentation. So you actually want to develop this system. So when we have a new virus in the next pandemic, you want to develop the antibody based on this. So actually, you have known the structure, but crystal structure is not available for the next the pandemic. So AI-based alpha default AI the defined structure should be utilized for the next pandemic. Have you ever tried that one? So what I want to say is structure is not defined for the next pandemic. So is it possible to have the same approaches? Have you ever tried that? Yes, I did. So I did not go far enough to have a simulations, but at the beginning of a pandemic, actually within 1 month, and we could have an interpretation of the structure. So Sungkyunkwan University, actually, for the first time as a private university, we have a cryo, the EM. Once we go into the cryo EM, daily 1 terabyte data is generated. We have a dynamic data and structural data, and both data those exist. So we are developing algorithm in order to have an interpretation. So within 2 weeks, within just 2 weeks' time, we have a two-dimensional and three-dimensional information so that we can go for the next stages. As I mentioned, we have alpha folder and other prediction and the information. So once we enter sequence information, and actually 93 and to 95% accurate information can be secured. So based on AI, we can have the create and the speed. So we have the question and the received in advance. So AI-based antibody development is done. So is there any the companies which actually develop new drug based on this? So the AI based antibody development and can shorten the time and the mutation and can be predicted. So as you mentioned, universal the antibody can be developed, which it can provide a solution to the rapid the mutation. So is there any case to develop the new drug? So based on the open innovation, and is there any case to have an open platform? So there are various trials to this approach. So could you share your experience about that one? So the antibody design was challenging. So because the CDR reason and the structure of that is not static, so we do not have the sequence information. We do not have a high amount of sequence information and structure information. But the well, the researcher, the Gibok, and in the United States, and actually provided some insight. And the company established by the researcher, listed on Nasdaq recently. So, those the companies provided some insight. So there is a great possibility down the road. Well, thank you for the presentation.
I would like to invite the next speaker. Dr. Gunul Lee will make the next presentation. He received the three degrees at Seoul National University. He served as a professor at University of Houston. Now he's the CEO of the Angel New Drug Designing Company. He's going to make a presentation about AI-based drug design, introduction, and applications on viral diseases. Nice to meet you. Thank you for the kind introduction. Thank you for inviting me to this meaningful symposium. I would like to thank the organizer. So the name of my company is in English AIDD, Angel Eye Drug Design. So I believe in Angel more than in AI. So I've been working as a professor at a university. Now I am working as a CEO of this company. And I was previously the representative of Korea Bioinformatics Society. Now I am also the CTO at Korea Quantum Computing, KQC. In the limited duration of time, I would like to talk about in silico computer-aided drug design. I receive a lot of questions regarding this, and many of you would like to know what this is about, but there are not many experts in this area. So I would like to give you some introduction about this area. I am one of those who brought this concept to Korea. And Professor Kim talked about the protein design based on computer system, and I work more on the in silico area using AI. All methodologies have pros and cons. I would like to introduce to you this concept and help you understand how to utilize this methodology as a guideline. Let me explain the history first. So at first, we didn't have the well-defined concept, but with the development of quantum physics, we came to have better prediction. Hansi is a famous pioneer. So they conducted thought first, but with the development of theories, we came up with quantitative structure activity relationships based on 2D, 3D. Now we are living in the era of AI. When it comes to pharmacophore modeling, for 2D, 3D, QSAR, they use regression analysis. There are many variables that explain the efficacy, so we make a kind of black box and see what kind of elements gives us the biggest efficacy. Deep learning is an advanced version which is related to this modeling. For pharmacophore modeling, so fundamentally, there are tens of hundreds of candidates. We need to find the meaningful one. The pharmacophore modeling is the first method we can use for this. We can identify the three-dimensional chemical, physical properties and structures of the molecules So this looks like a soccer ball. We screen this and get tens of thousands of data sets, and we run the data set in the computer and select and screen a meaningful data set. This is the black box. At top of the picture, we have the black box. And with computer-aided or in silico drug design, we can proceed with this process more conveniently. But many of you would not be familiar with this process and technology, so many of you would be confused about this. And docking is located in the middle of the picture, and as we have more serious calculation methods like quantum uh, computing. Basically, here Lipinski's rule is one of the example, and we have admin talks. When we develop a new drug, it is like winning a lottery ticket. So it is really difficult to begin and achieve success through a rational um, in a rational context. So we have to combine all wisdoms and knowledges we have. So at first we begin with massive chemical dB. We have to increase the probability, although the probability is very low at about 10% level. So it is important to have collective intelligence system in place. That's the source of your competitiveness. And pharmacophore modeling is done first. If there's a target you want, you need to figure out the active site. Also, you can make an mRNA-related protein, keep it from being born. You can also kill that virus, but usually we use small molecules to inhibit the activity of the protein. So activity site or binding site is the one we are targeting to inhibit the activity of the protein so that the protein will in the end die. So we eliminate this in this way. So in the site you want, physical chemical properties are identified based on the information. We use the tens of thousands of data sets in our database. And you can select thousands or hundreds of candidates, and then you move on to CAD with molecular docking. You might have heard of molecular docking often. This is the area of the professional experts. So the, when it is docked, the structure of protein is fixed, but when drug comes in, the proteins should react to this. This is very difficult and complicated process. Molecular structure is allowed to move a bit because it's better than nothing. That's why people use docking. But we can use AI these days. We can predict drug affinity. So the drug affinity can be estimated through AI quite quickly, but the precision or accuracy is not sufficiently high. So when drug comes in, the protein structure is adjustable, but this is not the area we can use AI completely. So you should be aware of the limits or constraints in the use of AI for this kind of molecular docking and dynamic simulations. This is very complicated and difficult. And this kind of research is very helpful for your mechanism research. So this is very important for research activities, and I am one of the experts in this area. There are not really many experts in this area in Korea, and I am really committed to this research. And another issue we face is in the case of docking, we have to evaluate the energy affinity, but AI cannot evaluate the affinity. So it doesn't have chemical, physical concept. And when there's a movement, the energy decreases and entropy increases. There's free energy. Sometimes A molecule docks better than molecule B. That depends on the level of energy and entropy. So when you calculate the free energy, when you select 2 or 3 drugs out of 100 drugs, it will be helpful, but it costs a lot to do this kind of calculation. So for ordinary researchers, what they can do at best is just docking. These days, there are a lot of methodologies with different names. Some people call this CAD, some other people call this modular modeling or computational chemistry. Intensive calculation is also called physics methodologies. There are not many professional experts in this area. But in terms of new drug, this is the most important field of battle. These days, there are many AI platforms adopted, so we can do things that we could not do in the past by using AI platforms. But in my opinion, we have constraints. This is the term I created, shallow screening and deep screening. Developing a new drug is the battle of screening. It takes such a long time for screening. And this is the area that is very difficult. In silico experts need to collaborate with drug discovery researchers. So, in terms of drug design and computational chemistry, AI revolution has not yet reached. This is what many people have thought about. As Professor Kim said, this is a battle of data. We have a lot of text and data in Google. We have to analyze the existing data and get what we want and respond to the situation. Chemistry or quantum calculation related content is not really provided sufficiently through the existing channels. When predicting the structure of molecule does not mean that we can develop new drug immediately. So that those two are two different stories. So identifying the structure of molecules does not always mean that we can right away develop the new drug. This is the computational biology and chemistry. So they involve very difficult calculations, and there's docking. And by using these computational methodologies, we can do the modeling and we can identify sequencing easily, but identifying the structure is not really easy. AlphaFold or Rosetta Fold is one of the most modernized and automated products. So our life became easier and more convenient. So the pioneers who received Nobel Prize in this area made our life much easier. And in terms of the dynamics, as time passes, it changes and moves a lot. This is an area where AI cannot cover completely. And for docking, it consists of protein ligand docking and small molecular docking. As for protein-protein docking, it is very difficult. We have a long way to go. So there are many pioneers, including the Nobel laureates, Marcinkovas and Berendis in Europe, Pastoid, who passed away, but when we learned about this technology, We used to say he is the representative of this area. And Mecca, he's my teacher, my professor. And as second generation pioneers, we have Shultan at UIUC. I am a minority researcher in this area. So when I published my paper on computational way of designing and developing new drugs, at first not many people paid attention to my paper. But nowadays, computational chemistry and biology received recognition. For example, those who developed quantum chemistry calculation methods received the Nobel Prize. Previously, there was a methodology called X alpha. So the developer of X alpha and the developer of XCFO, they did it in the 1960s. So I learned about their methods when I was at Seoul National University graduate school. And these two people, they developed the quantum chemistry calculation methods. John, John Pople and many other pioneers, they developed this computational methods. They were called semi-quantum computational methods as well. The Gaussian and games are good examples. They develop SD functional, density functional theory as well. So they opened the era of quantum chemistry, and we came to be able to conduct computational methods. So we came to be able to calculate the chemical and physical properties of molecules. And I was one of the Korean scholars adopting these methods 30 years ago. And they interact with proteins. And there are thousands of proteins. And the quantum computational methods deal with different number of molecules or proteins. And Marty Karpus at Harvard University and other scholars, they thought about how to further improve this. Previously they used Schrödinger equation, now they use Newton equation. The most important quantum chemical calculation is that they can follow the electrons, but now they drop electrons, but use atoms and identify the interaction between protein and drug. So 15 years later, in 2013, these scholars received a Nobel Prize in developing molecular modeling calculation methods. And then the scholars who used AI received the Nobel Prize in Physics in 2024. And David Becker and other scholars, they also received the Nobel Prize for opening AI-based protein design and structure prediction. They created various methodologies. They provide us with easier way of designing protein by using AI. Due to time constraint, I cannot explain all of the slides in detail. So when you first begin this process, you need to identify the structure of the protein. We have both conventional ways and new in silico ways. But as I mentioned before, understanding the 3D structure of molecules or proteins does not mean that you can right away develop a new drug. And we conducted 3 to 4 projects helping people to understand how they can use in silico drug design methods. This is one of the drugs used to treat COVID 19. So we can use AI system to find the optimal way to develop this system. So this drug is soluted in 30 minutes, so it has less stability. So we conducted a research to enhance the stability of the drug. So the relevant researches have been done by other researchers as well. So CIP proteins include the protein that degrade the drug. So SOM is the site of metabolism. So we identify the site of metabolism. There are various CI proteins. There are various types of SOM-H. So we specify these two, and we conducted substitution. There are various ways of substitution: single substitution, gender substitution. So we identified about 266, and we conducted docking to identify the ranking. In the case of Admitox, for each of the Admitox category, we can identify the ranking of the properties, and you can decide which drug you want to test first.
이런 거 내가 이제 사실 이런 것들은 파는 약을 가서 얼른 테스트해 보고 싶으면 그건 여기 병원 의사 교수님이시라든가
So some of you might want to test the drugs by using this method.
그 활용도를 알았으니까 그걸 뭐 대박을 예를 들어서 만들기 위해서는 사실 물질투구가 필요하면 구조를 만들어야 되거든요. 그래서 이제 그걸 제가
So depending on the case, you might need to file for patent for the way you found the properties of the protein.
그런 분들이 이제 나서서 이런 것들을 좀 할 수 있게 되고 그런 건 제가 어떻게 보면 그런
So here you need to also predict the compound synthetic accessibility. It requires navigation system, and if you collaborate with professional experts in this area, you can do your work in a more efficient manner.
두 번째 타겟에 대한 그런 연구도 좀 진행했고요.
And we conducted research on the second target.
그래서 아까처럼 이런 이제 270개 정도 조출한 다음에
We selected 270. We filtered them out.
또한 가지는 이제 지금 여러분들 아는 에이즈 타겟이죠.
And another target is HIV integrase inhibitor.
약 중에
Bictegravir is a well-known drug for that targets HIV.
이런 식으로 이제 엑스레이 구조가 나와 있는데
This way, it has X-ray structure.
지금 밀턴트 내성이 좀 강한 약물을 좀 찾고 싶어 하셨습니다. 저희 콜라보레이터가.
Our collaborator wanted to find a drug with strong resistance.
그래서 이걸 어떻게 만들까 좀
So we thought about how we can improve this. For mutant and wild types, we conducted MD simulation and identified the key points as time passes. And we also identify the affinity on a real-time basis. So we observed the situation, and so in this way we apply some changes. And we proposed our ideas and conducted in silico calculations. This is our study on mechanism. This is being done in collaboration with a professor at Jeonnam University. We applied point mutation, and the activity of the protein became very strange or different, and it has kinase function. And in the plant area, there are the three elements there, so we created a model. This is related to the binding affinity.
AI 쪽으로는 한 6, 7년 처음 우리나라에서 이제 AI 플랫폼 가지고 신약 설계하는 플랫폼을 만들어 봐라 하는 것과
In
관련된 프로젝트가
Korea,
꽤 있었는데
6 to 7 years
우리가
ago in
기업하고
Korea,
같이
we were asked to
협력해서 이제
create an
그걸
AI
하지 못했고요.
platform for new drug design, and our team participated
그다음에
in
이제
that effort.
그 알츠하이머 디지즈로 펩타이드를
And
이제 저희
for
금명대학교
Alzheimer's disease,
대학교에 계신 교수님이 찾아서 이제 이걸 특허를 피하기 위해서 좀 다른
in order to
패턴을 좀
avoid
바꿔달라 또는 이제
patents,
길이를 좀 줄여달라
we
그래서
were
저희가
asked
많은
to change the
연결되기까지
sequence
노력을 좀
or
했고요.
reduce some
연결되지
area
않고
in this.
그다음에 많은 수많은 MD 시뮬레이션을
So we
통해서
conducted various
그
MD
이로는
simulations
기류와 비슷한 정도를 찾아서
and found
그 길이를
structures,
줄였는데
similar
성공했고
structures, and reduced the length.
또 길이를 줄였는데 성공했고 또 이제 뭐 반감기를 늘려달라 뭐
And we
스탠다라이즈
were also
해달라
asked
해서
to increase the half-life of this
최근에 가장 어려웠고 한
element.
게 이제 프로탁 같은
Protac
복잡한 구조인데
design
이제 이런
has
부분에도
very
이론적으로
complicated
프로틴이 8개나 들어간다고
structure. It
생각이
has
돼서
8
굉장히 좀
proteins, so it's very
독보적인
difficult to
기술이다.
touch on this. It
내부적으로 생각하고
requires
있고
a really high level of expertise.
그래서 오늘은 이 정도로 마치도록 하겠습니다.
So with this, I would like to conclude my presentation. Thank you for your attention. Thank you for your presentation. Due to time constraint, we can take only one question from the floor. Thank you very much for your lecture. It's a simple question. We screen a lot of antiviral candidates based on protein structure. Previously, whole virus or a specific step of the virus or inhibitors of the virus were screened. But we couldn't find the receptor target, so it was very difficult. If there is a ligand, can you design pharmacophore based on the ligand? And is this possible? Do you have a strategy to design pharmacophore based on a certain ligand? Thank you for the question. So I also talked about it with other professors, but in reality, it's difficult. In MD simulation or docking, the drug appears to bind with any area that it touches. But interpreting this phenomena is really difficult. So you need to exchange your ideas and knowledge with professional experts in this area. In reality, what you said is not easy. But when there is a hole or the protein binds with the hole very easily, but in reality, it's difficult to design a pharmacophore based on a certain ligand. Thank you for your presentation. I would like to make a comment. So, although we have 2 more presentations left, by using AI, if we identify candidates by using AI, when we are targeting certain pathogens for infectious disease, we have to do the verification, verify the candidate. So Professor Park's team and Korea Pasteur team created a network to use a standardized method to evaluate and verify the candidate found for specific infectious disease. And your research based on AI could be very helpful for us in verifying the candidate materials. That will speed up the time. So this is just a comment. Yes, while listening to the presentation of Professor Kim, I thought that what I research will be complementary to Professor Kim's research. And doing this at a timely manner is important, and this is why collaboration is important. I also would like to look forward to a future collaboration with you. Thank you very much.
Session 1, so let's take a 10-minute break and we're gonna resume at 11:20. 네, 여러분 이제 두 개 발표 들어서 중간까지 왔는데요. 잠시 10분 쉬어가겠습니다. 11시 20분에 다시 뵙도록 하겠습니다. 감사합니다. 네, 쉬는 시간 중에 잠시 안내 말씀드리겠습니다. 오늘도 정말 많은 분들이 함께 네, 저희 1분 후에 시작하겠습니다.
In 1 minute, we are going to begin the session.
The session in 1 minute, so please return to your seats. So, okay, welcome back. Welcome. So, we're going to pick up where we left off. So, two more presentations are waiting for you in the session one. And just like the before, we're going to give the floor to our grade 2 co-chairs, Dr. Juyeon Lee and Professor Man-sung Park. Please give them a big hand. 여러분, 큰 박수로 다시 한번 세션 시작하도록 하겠습니다.
So let us continue. So actually, next speaker is not able to be with us in person, so we are going to be connected through Zoom platform. So let me introduce speakers first. So, well, we are going to invite Dr. Evelyn Georgiev. So he is from Vanderbilt University. So he is a professor of that university. Antibody therapeutic research. The effort is led by the Mr. Evelyn Georgiev. So in the year 2009, from Duke University, he received a PhD degree for the computer science. AI-based antigen development study is led by him. So he's going to share his insight about that.
Jeff, are you ready?
Yes, hello.
Can you hear me?
Yes,
Okay, so please go ahead with your presentation.
thank you so much for inviting me. And it's a great pleasure to be able to present some of the recent work in my group related to AI platforms for monoclonal antibody discovery. And I'm just very sorry that I couldn't be there in person, but hopefully this shows exciting stuff that we've been doing over the last couple of years or so. So I'm sure for this audience, I do not need to introduce what monoclonal antibodies are, but they have really transformed how we generate new therapeutics, a variety of different diseases. As you can see, over the last couple of decades, there has been an explosion of antibody therapeutics that have been approved for use in the clinic. And this therapeutics related to a variety of different diseases, cancer, autoimmunity, number of other disease targets as well. This market is projected to grow to over $1 trillion over the next decade or so. So this is a very important area of research, and we should really try to optimize the way we discover antibody therapeutics and lead them toward the market. So this is what I'm focusing on over the last few years. But the traditional antibody discovery process is like searching for a needle in a haystack. So basically, if you imagine that all of the possible antigen targets are represented by this collection of black dots here. So each antigen or a family of antigens is one of the dots, and then dots that are close to each other are maybe antigens that are related to each other. So the first step in the antibody discovery process is to identify what the antigen targets are. Once we know what these antigen targets are, then we typically have to get access to samples that may have antibodies against these antigens. This is typically either human samples, infection or vaccination, or animal samples, or some other source. Then what we need to do is to search for the antibodies that are specific to the antigen targets. And for human antibodies, at least, we need to know both the heavy and the light chain sequence information in order to be able to make these antibodies in the clinic. We need to make sure that the antibodies have the desired target antigen specificity, and then we can down select for functional phenotypes. Now, this process has led to a lot of success, as you saw on the previous slides. There have been quite a few antibodies that have been approved in the clinic, but it is also associated with a lot of different challenges and disadvantages. This process is inefficient, which leads to wasted resources and low likelihood of success. It requires access to often rare and difficult to obtain samples. It can take months to years for the initial discovery phase, and this process is even less likely to succeed for challenging targets. It also requires specialized expertise and expensive equipment, which severely limits who can do the discovery. So because of all this, we started thinking about what we can do given our expertise in computational immunology, and specifically whether we're able to develop methods that are based on artificial intelligence to improve on how antibody discovery is performed. Now, we think that AI can be very helpful in this area because it can lead to much more efficient discovery of antibodies with precise phenotypes. It can be applied to otherwise intractable targets. The process would not require access to rare samples, specialized equipment, or expertise. And in reality, it will democratize the discovery part of the process, so anybody in the room on Zoom can actually do antibody discovery for whatever target they're interested in. So this is our motivation, and we have been doing this for a few years now. We have published along multiple different directions in this area of AI for antibody discovery, but to highlight today some of the more recent work that we've done that was published in the journal Cell a few months ago. So in this specific formulation of the problem of AI for antibody discovery, what we're really interested in is to try to design antibodies from scratch. And so the idea that, or the question that we're posing is, can we start with an antigen sequence of interest, as in the shown here on the left, and obtain a pool of antibody candidates as shown here on the right? So what we're really asking is, can we go from antigen sequence to antibody sequence? Now, to develop any type of AI or machine learning algorithm, of course, we need good training data sets. But unfortunately, in the case of antibody-antigen interactions, those training sets are actually quite limited. First, as I mentioned, antibodies, at least human antibodies, require both heavy and light chain information. And a lot of the data that's out there is limited to either only heavy or light chains, but not the paired heavy light chain information for antibodies. Even for the paired data, for much of the remaining data, there's really no antigen specificity information available. And whenever antigen specificity information is available, it's restricted to only a handful of the most frequently encountered pathogens. So this is not an ideal data set, and that's what led us to think that maybe we can also help with the data set generation to improve the likelihood of succeeding for the AI-based methods. And so we have another platform that we call LibreSeq, or Linking B-cell Receptor to Antigen Specificity Through Sequencing, which is an experimental platform for high-throughput antibody discovery against large antigen libraries. So what we do there is we can screen a given donor sample, and we can obtain a very high-resolution mapping of how well each of the given B cells that we discover recognizes a set of antigens, as shown in the schematic here. Now, we can do this for many different donors, many different samples, and do this for a large set of antigens at a time, with the goal of creating a large-scale antibody-antigen atlas of unparalleled size and diversity. So this is what we want to do, and this is what we're working on, and hopefully within the next couple of years, we will have such data sets. But in the meantime, we thought it's better to not sit and wait until we generate the data, but see what we can do with the existing data. So to address this question, what we did was we compiled the publicly available data, as shown in the different databases that are highlighted here, as well as our own published and unpublished LibreSeq data. And this led to the generation of around 15,000 training examples of curated data from the publicly available and our own data sets. But unfortunately, 2/3 of them were related to SARS-CoV-2, and for the remaining data, these examples were distributed between only a handful of the most frequently encountered pathogens. And so this is not an ideal data set, neither by size nor by diversity, but we thought, well, let's just see what we can do, and maybe we can generate some exciting results, even though there's a lot of limitations with the data. And this is what we tried to do. The next question that we asked was, what kind of algorithm should we actually try to develop for this type of problem? And we have several different algorithms, but the one that I will be presenting here is based on generative AI, which is large language models or protein language models. And if you think of any type of chat model that's out there, basically it's a very simple process of asking a text question and receiving a text answer. And this is really ideal for our case because protein sequences or antigen sequences are just like text, and this can be our question or prompt, and then the response that we're awaiting is the antibody sequence. So the question that we ask is an antigen sequence, and the answer that we expect is an antibody sequence that should be specific to that antigen sequence. And more importantly, again, if you're using any of the chat tools that are out there, if you ask the exact same question multiple times, more likely than not, you will receive a different answer every time. Now, sometimes it will be an answer that's kind of similar to something that you already saw, or in other cases, you may get a completely different answer. And this is very important for us because we want to generate a diverse pool of antibody candidates. We do not just want to generate one antibody every time we ask or pose the question of that specific antigen. We want a pool of candidates that we can down select from later on and then leading to our top candidate. And so with this in mind, we decided to develop this algorithm, which we call MAGE, or monoclonal antibody generator, which is the first version of our algorithm. We have more versions and more architectures that we have developed since then, but I think this really illustrates well some of the applications of this type of AI for antibody discovery. And so what we did was we used the pre-trained protein language model, and then we fine-tuned on the antibody antigen data set that we had created. And then we developed the MAGE algorithm to be able to generate antibodies to antigen sequences that we provide as input. And to be able to validate that this AI algorithm actually works in practice, what we decided to do was to apply it to 3 different targets so that we have diversity of representation within the training data. So SARS-CoV-2 receptor binding domain was the first target that we selected, and there were around 3,000 training cases for SARS-CoV-2 RBD. Then we selected respiratory syncytial virus, F protein, with around 300 training cases. And finally, we had influenza H5 hemagglutinin with 0 training cases with that exact hemagglutinin in the training data. And I'll explain this in a moment in a little bit more detail. But first, I want to highlight that when we applied MAGE to all 3 targets, then we generated antibody candidates, we down-selected a handful of them, we produced them in the lab, so experimentally, wet lab, as recombinant monoclonal antibodies. And for all 3 cases, we actually had good hits. So this is just a schematic way of representing hits. Darker blue colors, each row represents an antibody candidate generated by the algorithm, and darker blue colors represent stronger recognition of that AI-generated antibody, that specific antigen target. So for all 3, we had really good candidates. But just to show you how this process actually works, I will illustrate this first with the RSVF protein. And the specific RSVF protein sequence that we used is shown here. So this is really the input to MAGE. You don't do anything with structure, just use the antigen sequence. So we input that antigen sequence into MAGE, and then we asked it to generate antibody sequences. And what we did was we did this 10,000 times. So we generated 10,000 antibody candidates, and it took around 15 seconds per antibody. So looking at these 10,000 candidates, the first question that we asked was, we postulated that using large language models, protein language models, generative AI can generate a diverse set of candidates. But is the reality, or did we actually generate just one antibody? And to answer this question, we looked at the combinations of heavy chains and light chains for the antibodies that were generated by MAGE. So each row here represents one germline gene heavy chain, and on the x-axis we have the light chains. The heat map represents how frequently a given combination of a heavy chain and a light chain was used for the antibodies that were generated. Darker colors mean higher frequency of generation. And so you can see that there are some combinations like IgHV1 18 with IgKV2 30 that were more frequent than others. But overall, there was a distribution of combinations that spanned a large variety of heavy chains and light chains, indicating that indeed the algorithm was able to generate diverse antibody sequences. So not just one antibody sequence, but a pool of antibody sequences that are different from each other. So that was the first question, but then the second question that we wanted to ask that is also very important is, okay, we have different antibody sequences that are generated by the algorithm. So compared to each other, they're different, but are they actually different from what is in the training data, or is the algorithm simply reproducing what it saw in the training? And so to address this question, what we decided to do was to take each of our age-generated antibodies, AI-generated antibodies, and then compare them to the training data, and then identify the closest match in the training data to our generated antibody. And the results are shown in this plot here, with the specifically for the heavy chain, with the x-axis showing the minimum distance of anything in the training data to each of our antibodies, and then the y-axis is just the density, how frequently that minimum distance occurs. And you can see the three different colors for the three different antigens: gray for SARS-CoV-2, blue for influenza, and orange for RSV. And you can see that for all three cases, we had, on average, more than 10 differences in the heavy chain compared to the closest antibody in the training data. And in the case of RSV, this actually approached 20 differences. So this is very substantial number of differences for the antibodies, confirming that we were able to, the algorithm itself was able to generate relatively novel sequences that are not simply seen in the training data, but that are novel and different from what is already in the training. But of course, just looking at antibody sequences is not enough, and we need to make sure that they're actually doing what they're supposed to be doing. And to test this, we did a couple of different assays. The first one is binding assay. This is an ELISA binding assay with the concentration of the antibody shown on the x-axis and the strength of the signal shown on the y-axis. Good binding. Each antibody that's tested is shown as a different curve. And for good binding, we have the curve shifted to the left. And you can see here that we have really good binding for several of the antibodies generated by MAGE when we tested them for binding to the RSV F protein. Then, in collaboration with the group of Giuseppe Salto and Ted Ross at Cleveland Clinic, Florida, we were able to test neutralization of virus. So binding was the first step, but then we wanted to also test functionality, and we did this by testing antibody virus neutralization. So again, concentration of antibody on the x-axis, and then percent neutralization on the y-axis. And you can see that there's actually at least 3 antibodies that neutralize virus quite well. So this is really exciting because now we see that we can actually use AI to generate novel antibodies that can neutralize virus. But not only that, we've been able to obtain a high-resolution cryo-EM structure of 2 of these antibodies with the RSVF protein. And this was work done by Jason McClellan's group at UT Austin. So in the structure, you can see 2 of these antibodies, one shown in different shades of blue, heavy chain in dark blue, light chain in light blue, and then we have another one 3301 shown on the bottom here in light green and dark green for the light and heavy chain. And then the RSV-F trimer is shown in 3 different colors, pink, red, and magenta or so, or I guess that's more like salmon. So what's very interesting about this particular antibody 3301 is that as far as we know, this is the first structure of a fully human antibody that binds to this specific site of RSVF. Not only that, but this antibody is very interesting because it exhibits around 20% mighty hypermutation within the heavy chain, which is exceptionally high, and then it is around 10% different from the closest training antibody sequence. So this is a novel antibody, fully human, and it binds to a site that previously had not been visualized using high-resolution structure. And so this was the the story, but then what I think would be interesting for this audience as well, as it is application of MAGE to the flu hemagglutinin from a virus that arose in 2024, highly pathogenic avian flu outbreak in cows in the United States that also transferred into humans. So there were multiple human cases as well. And so what we wanted to ask was, can we apply MAGE to this specific case because there were no other hemagglutinins with that exact sequence in the training data. There were other hemagglutinins that were related, so other H5 hemagglutinins, but nothing with that exact sequence. So we were asking, well, can MAGE extrapolate based on the training data that it learned on related sequences and generate functional antibodies against this novel sequence? And indeed, when we use MAGE to generate antibodies, then we down selected and produced them in the lab and tested them, showed that we can generate functional antibodies against this novel influenza hemagglutinin. The graph here shows neutralization data similar to the previous slide, and this is summarized in the heat map here as IC50 values. The darker colors mean stronger neutralization. Rows represent antibodies, columns represent viruses. This new virus that I'm highlighting is shown as the first column here, and you can see that all of our antibodies that were tested, along with this positive control, show good neutralization of this virus. But not only that, but they could also neutralize other viruses from H5N1 as well as H1N1. So we were able to generate broadly neutralizing antibodies using AI against this new influenza virus. And what makes this really exciting is that this is a potential real-life use case scenario where we can do ultra-rapid pandemic response to a new viral variant that has just emerged that may be different from what we have seen before. So where are we taking this next? We really want to build an integrated platform that combines both wet lab and computation, using our LibreSeq technology, using our MAGE technology, as well as a variety of other computational algorithms and platforms that we're developing that will allow us to do targeted optimization, generation and optimization of antibody candidates for any target of interest. We're not quite there yet. We really need to continue working on database generation, to train our algorithms, develop new architectures, and so on. But we're very excited with the current preliminary steps that we have been able to accomplish. And we have a number of targets that we're currently pursuing. Of course, we're continuing our work with infectious disease, but we're also targeting a few other disease areas that we think we will be able to contribute to. And so with this, I want to thank everyone in my lab for doing all this really great work over years, as well as our collaborators and our funding sources. And I thank you for your attention, and I will be happy to take any questions.
Thank you for your wonderful talk. Now we will now open the floor for question. Yeah, okay.
Thank you very much for your very interesting and predicted insightful talk. I have one question. I already this question in previous to the talker. Given a sudden the pandemic situation by disease X, based on the actual based on the current technology, what if of antibodies generated by AI, do you predict will actually be functional?
That's a great question. And I think that there's two parts to that. So a lot of the diseases, a lot of the pathogens that, you know, have emerged over the last couple of decades, you can think of as related to pathogens that we have already seen. That's true for coronaviruses, of course, the highly pathogenic coronaviruses. Of course, we've had endemic coronaviruses for much longer than that. Similarly, with influenza, highly pathogenic influenza, there are related viruses that the human population has seen. And I think in those cases, we can have really good confidence that the antibodies that we generate will be functional. If it's a pathogen that we have not seen, nothing related to this has emerged in humans before, and there's no training data for that, the success rate is probably going to be lower, but I do think that it will be possible to fairly rapidly generate antibodies that are successful against this pathogen using the technologies that we have currently.
Okay, okay, go ahead.
Hey, great talk. This is Jimmy Gallagher at Houston Methodist Research Institute. I have two quick questions. One, given that the training data is largely on memory B cells, what fraction of those differences that you see are due to germline reversion? And two, you made 10,000 antibodies to these three targets. How many of them did bind?
So to make sure that I understand your first question, are you asking out of the differences when we compare between training data and the generated antibodies, how many of these differences, what fraction of these differences are due to germline reversion?
Yes.
Okay. Yes, we did this analysis, and what we observed was that in a number of cases, we are generating mutations that are not in the training data. So there are somatic hypermutations that it's not germline reversion, but it's different mutations that are not in the training data. So that was exciting to see because we're not simply reverting to germline or looking at consensus sequences. And sorry, what was your second question?
Out of the 10,000 that you built to the three targets, how many bound?
Yeah, we didn't test 10,000 in the lab just because that's way too prohibitive. I think it varied between something like 20 and 25 antibodies that we made for each of the targets. And I think the numbers were between 20 and 40-something% success rate.
Okay, is there another? Yeah, okay, yeah, go ahead. Thank you for your great talk, Evelyn. I'm Song-Yeol Ko from International Vaccine Institute. So I have a question about the immunogenicity. So your designed antibody is not a natural one, but the artificial one. So I'm considering if your designed antibody is immunogenic or not during your design of that antibodies. And the second question is kind of technical one. So you may not have the answer, but for the liver-seq assays, so in that technology, it's usually recombinant protein is as the antigen. I'm wondering if the virus particle or virus-like particle can be used as antigens.
Yeah, so to answer your second question first, yes, you can use virus-like particles as the antigens in liver-seq. There was a recent paper that was published, I believe, with virus-like particles, and we have done, not by our group, by someone else, and we have done VLPs as well that, or similar work that we have not published. So it is possible you don't have to use recombinant antigen, and we're developing new versions of liver-seq that will allow us to do much deeper screening compared to what we can do with recombinant antigen. Then to answer your first question, the antibodies that we generate, we check for humanness. So there's computational algorithms that allow us to look at how human the sequences of these antibodies are. And for the most part, they're fully human antibody sequences. If we're developing this forward toward the clinic, of course, we can perform polyreactivity assays and similar assays to make sure that autoreactivity is well, that there's no reactivity against self-antigen.
Is there another question? Okay, just due to time limitation, we will have to end the questions here. Okay, Dr. Evelyn, thank you so much. Let's meet in Seoul next time. Good night.
Thank you, I appreciate it.
예, 다음 연장.
Next speaker is Professor Namsik Han. He is serving dual position. He is a professor at University of Cambridge, and from last year, he's been serving as the professor of IBS, Quantum Informatics Department at Yonsei University. Today, he will make a presentation about quantum-inspired interpretable network AI for therapeutic discovery. Good morning, distinguished guests. I am Namsik Han. Thank you for inviting me to this wonderful symposium. Before making a presentation, before I forget, I want to share with you two things. The previous speakers talked about structural biology a lot. For the past 20 years, I served as a professor at University of Cambridge. This is the home to structural biology. It has world-renowned institutes as well. The Nobel Prize laureates in his slides were from University of Cambridge. Maybe half of the Nobel Prize laureates that you saw in his presentation were from University of Cambridge. In July, at the University of Cambridge Research Institute, there's a symposium about new drug discovery. It is Academia Industry Joint Symposium. If interested, please come visit us. Please come to the symposium. I will be there, so I am ready to welcome you. So for any information you want, please come. And if you want to meet me at a place that's close to your home, please visit Yonsei University. In April, Professor David Becker and the other Nobel Prize laureate will visit our campus in our forum. So please, it's actually after 2 weeks. So please come to visit us and the Nobel Prize laureates in our forum in Yonsei University in April as well. Now, I'm going to talk about new drug discovery based on quantum-inspired AI network. First, let me talk about what we began in 2022. When COVID virus appeared, as a scientist and a researcher, I was not an expert in infectious disease, but scientists and researchers made a great contribution to controlling the spread of COVID-19. And we began the IDOI, and we try to figure out how we can use our know-how and technologies for tackling COVID-19 threat. And we published a paper in 2021 or 2022. This is the activity summary. First, we wanted to discover new drug. If we find a totally novel target, it would be good, but that's very difficult. So we needed to take the shortcut. We reviewed approved drugs and used various AI methodologies. And if you look at the process here, among 2,000 approved drugs, we shortlisted 200 drugs, and then we shortlisted the 200 again to 46 drugs. And our labs conducted thorough verification of the drugs, and we validated the drugs, and the final outcome is that we have 2 validated drugs. Due to time constraint, I cannot explain about all the processes in detail, but let me first talk about the important parts. Not only our lab, but also many other labs of scientists participated in this kind of research. This is the paper published in Nature in summer in 2020. In June 2020, this paper was published. At that time, the author was not the infectious disease expert, but due to COVID-19, the authors wanted to use their own talents and know-how. So they found a lot of elements here. The red dots here refer to the outcome. In SARS-CoV-2, this is NSB number 1 or spike protein. The red diamonds refer to each part of the virus. And you also see here the proteins. So they found the proteins that interact with the SARS-CoV-2 too. And in the bioarchive, we saw this literature again. Instead of directly interacting protein, they found what kind of proteins are collected and mutated inside the human body 2 hours, 6 hours, and 24 hours after infection with this virus. And in August 2020, this information was published. And in the middle of January, we conducted an idea meeting. We do a lot of computational work using AI. So we downloaded the existing literature without peer review. So we had to verify the quality of the data. And we chose the 2 most promising data sets. And Dr. Woo Chong-hwang and Dr. Mea McMahon worked with me. And after becoming a PI in the UK, I was not allowed to go outside home. So during COVID-19 period in the UK, I did coding myself and I stayed home while studying and researching a lot of things. And directly interacting proteins, the number was 583. These are the proteins that first initially interact with the SARS-CoV-2. And there are proteins that are expressed in our body after infection. These are the causes. And many biologists want to know what kind of mechanisms take place in the middle of the process. Through what kind of mechanism this phenomena takes place. We therefore used our AI methodologies. And we thought that there's a hidden layer between the 2. So by using AI methodology, we constructed the hidden layer, created a network. Now it looks very beautiful on the picture here. But there are 10,000 proteins included. So in just bare eyes, it's almost impossible to see a detailed mechanism. So we adopted network algorithms to uncover key pathways. Through the key pathways, directly interacting proteins are accumulated and they are differentially expressed in our body. And through protein-protein interacting network, our protein-protein interaction network is a bit different, but in this wide network, we simulated each of the 2,000 approved drugs and found the promising inhibitors. Finally, we shortlisted 2 inhibitors. And 6 hours after infection and 24 hours after infection, there was the biggest change in the expression of protein. As you see here, the 13,000 nodes, and there were a lot of interactions among proteins. So this is not really informative at this level. And then we used various network algorithms. The blue and yellow colored pathways are important key pathways. It's difficult to explain about all the details, but so please refer to this paper later on if you're interested. Then what is network analysis? This is PPI network. There are about 160 proteins. It looks complicated, but if you open your eyes wide, you can see there are a lot of meaningful protein interactions, but we have over 10,000 proteins. So if the number of protein increases 100 times, you cannot do this work with your bare eyes. This is why we use the random walk algorithm. In the genius scientist Alan Turing from Cambridge created this method. So he's famous for his Turing machine. In the 1950s, he developed that Turing machine theory, and we're still using this theory. It applies not only to biology. So after the training, we can get the score for each protein. For 160 proteins, we can get a score for each of them. So it ranks the protein based on the score. So we can see the top first, and the protein ranked at the bottom as well. This is applied not only to biomedical area, but also to the recommendation algorithm of Netflix. The Netflix recommends you certain drama or movie based on what you watched. It recommends movies in which the actors or actresses whom you saw in the other movies appear. And when there are newly released movies, the Netflix runs the recommendation algorithm and gives you the customized algorithm. The same logic applies here as well. So it is also used for LinkedIn, the human networks, and recommending people you need to know. Amazon also uses the algorithm, recommending products that you might prefer. And we use this for identifying the proteins and inhibitors. Below, you see another network with different colors of red and blue. The colored nodes refer to important pathways. As for network algorithms, we have different types of algorithms. These are representative ones. Random walk with restart. It needs a start node, and it is very useful for finding a node that shares the influence with the other node mostly. And pathways here crosstalk a lot. So if you kill this pathway, you cannot see your expected impact. Another pathway still remains alive. So this is just an example. So if you use random walk, so we find nodes that are close to the starting node, and for RWR, you can find proteins with closer relational network with the other nodes. So I'm talking about the network algorithm for long with lengthy statements. This is because I want to explain about quantum walk. This is a very useful tool. Random walk means that you walk across the network randomly. For computer, it's not hard to walk across the network randomly. It means that it just decides whether to go right or left on its own, and it follows the path with the highest probability. So we find the node that is most likely to be visited by this random operator. And there are some constraints. By theory, through random walk, we should find the answer, whatever the situation, but the larger the network, it doesn't easily find the answer, and it continues to explore limited scope of areas in the network. So in order to overcome the constraint, we came to apply superposition concept in quantum theory. We apply this superposition concept. So when the walker begins to walk around the network, we use spot position, and the walker can locate different nodes at the same time. This is AI quantum hybridization. At the first phase, it was very difficult, but it took 1 year until we finally achieved success. And then we published the relevant paper as well. To help you understand this better, let's look at this London Tube map. London Tube map is one of the world's oldest map. As you see, the UK has achieved a lot of things for the first time in the world. This is do you remember the London downtown street? The background of Sherlock Holmes movie? So that's the location where the first subway of the UK was created. In this situation, you don't need the network algorithm to find the way or path. But from our perspective, data is very important, especially when it comes to AI. So proteomics or transproteomics or business. So multi-omics sequence data is often used in our work. In 2000, when Human Genomics Project was completed, sequencing cost was about 90 billion Korean won. The price is much lower this time, but in 2000, in the past, we had to spend a huge amount of money. So this is the level of map we will be able to create in the very past due to the constrained budget. But it gradually expands as time passes. Like when it became 1950, the map develops and expands in this way. So the data traffic rapidly increased and the cost decreased significantly. So by using multi-omics structural data form, we can get a lot of meaningful information, like super large PPA network. In this situation, random walk uh, has a lot of constraints. This is why we need quantum walk. This is the subway map of Seoul. You're familiar with this. We are located in Dongdaemun. If you want to go to Daejeon through KTX, you have to go to Seoul train station. You have to take line number 4 subway station. Then you will get off at the platform of Seoul train station. You forgot to bring your bag. Actually, you left your bag in the subway, the train. Then you have to call the police or you have to call the insurance company. But anyhow, you have to find uh, your bag again. So you will have to randomly visit the subway line number 4 stations to find your bag. So let's say that we have multiple people, like 100 people. You have 100 friends with you. They can help you find your bag by visiting line number 4 stations altogether. Then what would you do? Maybe 3 persons or 4 persons will visit several stations around the subway number 4, and some others will visit stations that of the subway lines that overlap with subway line number 4. So you may use different methodologies to find your bag by using the 100 people, 100 friends you have. So in this way, you can more quickly find your bag, and this is the way we apply to the new drug discovery, the concept of quantum walk. Then biomedical data is what we're going to use, so we have to see if this concept works. Here again, Dr. Woo Chang-hwang and Dr. Chesop Park worked very hard and they published a paper on this concept. They studied long COVID. The most important element here is that the starting node is very important in this algorithm. SARS-CoV-2 and in other types of infectious disease, it begins with SARS-CoV-2, the virus, and there are over 300 interacting proteins. We have PPA network, and there are different layers of network. So there are shallow nodes and deep nodes. And we applied quantum walk and random walk. And as you see in this network, if you look at SO, at deep nodes, you see more proteins expressed. And there are differentially expressed proteins, and there are more differentially expressed proteins in the deep nodes. The ground truth is that in the network here, there are blue dots. Blue dots refer to the blue dots areas have been researched a lot by long COVID or COVID researchers. So the blue dots refer to the ground truth data. They are important markers. We ran quantum walk and random walk, and AI algorithms found a lot of dots here, but not much in the deep nodes. In the case of quantum walk, they showed better search outcomes in the deep nodes. Then we should find some meaning, a significant meaning from what we found through quantum walk and random walk. So conclusion is that what is visible here, these lines refer to interaction. The gray lines and black lines refer to shallow nodes and deep nodes. This is shallow nodes, this is deep nodes. Quantum walk has a lot of interactions taking place here. Here, there's almost no interactions. And the functions, neurosystem or glucosylation, they function more in quantum walk. They're closely related to long COVID. And combination of these two, and how is this related to brain or neural system? There's celiac acid, and glucosylation is connected to this, and SARS-CoV-2 is connected to celiac acid. It can bind with the celiac acid and damages the brain cell, causing it damaging the membrane. And damages the plasticity of the synapse. So through this research, we found hidden links. Then among top 10 targets, there are TP53, which is a famous target related to cancer. This was detected through quantum walk. 3 out of 10 here belong to the one protein family group. So what they do is to regulate ROS metabolism. And at network level, this is schematic network. At PPI network, you can see this. So we zoomed in to the network surrounding CIDS 123. There is an agent that regulates the calcium ion, and dysfunction of mitochondria affects the functions of the phenotype here. And VDAC1 is a protein we should target. This is what we found through the research. And cannabidiol is one of the drugs that are known to be able to regulate this VDAC. And according to an independent study, it is likely to affect COVID-19. In summary, quantum walk provides a deeper exploration and of the global structure. It, it enables us to search for the target in a more precise way. So if you use quantum walk, you can identify new drugs that you were not able to do in the past. Now, we have a few more slides. Then why do we need quantum computing in drug discovery? AI is, of course, doing very good here, but why do we need quantum computing? According to Boston Consulting, McKinsey, or Deloitte Consulting, they all say that quantum computing, pharmaceutical industry is one of the industries that is going to be the first adopter of quantum computing because quantum technologies along with AI are likely to be applied to various areas, not just climate prediction. So according to a lot of consulting agencies, pharma is expected to be one of the first industries to adopt this. And quantum computing will bring about a great economic value. Although we don't know how long it will take, whether it will be 50 years or 100 years, but according to this data, after about 2030, quantum computing will become commercialized, and that will accelerate our drug discovery efforts. And so you can see the potential here on the slide. And multinational pharmas are already working with quantum startups as well as IBM and Google to apply quantum computing to their drug discovery efforts. So far, there have not been many cases where quantum was used for functional analysis, and our research is one of the world's first research that applied quantum computing, so we take pride in this. At Yonsei University, we have Yonsei Quantum Complex. It has IBM Quantum System 1. And Yonsei Quantum Complex is not just a simple hardware, but we have a department of quantum study at our university, and the professors are experts in quantum theory. They also use quantum technologies in their research activities. This is an example. Just as the previous speaker said, the Alpha H was mentioned during his presentation. It was a DARPA program. Now, that was under the Biden administration. In Korea, we are also making efforts for innovative new drug discovery efforts. From last year, Yonsei University was designated as the lead institute to proceed with the development of next-generation intractable cancer treatment through quantum-based drug discovery platform. So we should take the lead in this using the quantum structure in a new drug discovery. We are utilizing the drug discovery expertise of University of Cambridge, and we are also working with pharma companies as well as labs that are working very hard using quantum methodologies. So we have formed this kind of industry academia joint research system. For the past 20 years while living in the UK, I saw many institutes. I wanted to see a lot of institutes in both countries co-working with each other. So if you're interested, please contact us, and we look forward to cooperation with you for the future. Thank you very much for your attention. Thank you for your presentation. In Korea, there are many people with different perceptions on drug discovery based on quantum technologies. And while listening to your presentation, I thought that the quantum technologies could accelerate the new drug discovery process. We have President Chong Yong-ki there who has a question. Yes, thank you. I am Chang Seung-gi from Korea Pasede Research Institute. For various infectious diseases, we are conducting research. We have a good screening system as well. And regarding various viruses, we have gained very important tips as well. Can you identify targets through the quantum technologies? This is the first question. And if that alone is insufficient, what other methods can we use to find target ID? Thank you for the question. Simply put, the answer is yes. It is possible. So if you look at, so we first target ID validation, and if we achieve the hit, then we can achieve success. But my answer for your first question is yes. So our new project has parallel in its name. So we use quantum-based parallel drug discovery platform. Doing this research in the crossways or parallel ways is important. As we move on to different stages in drug discovery, it's like overcoming the valley of death. So we need to combine AI technologies and further advance translational research and increase the probability of success. Did it answer your question? Please use your microphone. What we need is a budget. What we need is funding. And Director Nam. Thank you for the good lecture. So I thought I would get the first chance to ask you a question. It's a joke. Thank you for the interesting presentation. Before joining the National Institute of Infectious Disease, I was very interested in figuring out the side effects of mRNA vaccine. I wanted to figure out the mechanism of side effects caused by mRNA vaccine. In order to do this in the WebNet, if I ask you to let me know the key pathway for candidates, what kind of data do you need? Not the funding. So what kind of data do you need if I want to find the key pathway? Regarding toxicity, the phase 1 is about toxicity. It's very important. But unfortunately, whether it be AI or quantum, we need a huge amount of data. Especially for toxicity, it's very difficult to get toxicity-related data. Pharmaceutical companies or institutes have data about toxicity, but both in Korea and in the EU, the toxicity-related data is considered a precious asset of the private company, so it's difficult to get the data. But if you promise me to provide us with the data, then one of the data we need is multi-omics data. Time series data is required. Time series data that enables us to compare the situation before and after a certain point will be necessary. So we could do some basic training and we could use the data about virus that is likely to mutate. So our lab engages in multi-omics. So the data we need is multi-omics data and longitudinal data as well. Another question? I am from National Institute of Infectious Disease. Thank you for your lecture. So, based on the database accumulated so far, we conducted a lot of research. And it would be good if we can use the existing clinical data, but if we see the emergence of new pathogens, the amount of data set it decreases, and there are a lot of things we don't understand perfectly, whether it be vaccine or any other things, or for rare diseases. So what kind of approach do you think we have to take for new pathogens or rare diseases? So if it will be good if we can create a good quality foundation model. If the foundation model is good, we can make the good prediction for any emergence of new pathogen. And if you use quantum and LLM based model, you can create various structures and conduct some simulation. But LLM has its limits. LLM generates similar forms of information. AI is somewhat lazy, so you need to use quantum. So quantum-based generative model gives you more variety of information. It could help you predict unexpected mutations. Do you have another question? I have a lot of questions, but let me ask just one last question before lunch. In the infectious disease area, we developed vaccine, but in the case of SARS-CoV-2 and influenza, we see continuous mutations taking place, the preventive effect declines. In order to overcome this issue, many researchers are studying how to predict the mutations. Amino acid that construct a protein has preference if there's change in the major the protein. So from the evolution perspectives, we can select the proper protein, or in the protein-protein interaction, it is really difficult to predict which protein is going to be mutated or which protein is going to have what kind of preference. So do you find any preference? In the Science Advanced paper, there's a similar content. Spike protein and LSB, non-structured protein 8, they have a lot of crosstalks. and it is closely related to biological mechanisms. 6 months after publishing the paper, NSP number 8 gives a big impact on replication or metabolism. From amino acid level mutation or preferable combination, these are the things we didn't hear or find in the past, but I think it is possible to use quantum to find the evidence for the protein mutations. Thank you for your presentation.
Thank you very much, and thank you, Cheers. Thanks to your wonderful moderation and the great contribution from all speakers, we just successfully conclude the session 1, which is morning session. This is a high time to take a lunch, so let's take a lunch break until 1:25, and we're gonna resume right here from 1:25. And for those who are taking part in offline event, please remain seated until you get your meal service. Thank you. 네. 네, 다시 한번 식사 안내해 드리겠습니다. 귀빈분들은 스튜디오 1번에서 따로 모시겠고요. 또 참석해 주신 모든 분들 자리에 잠시 앉아 계시면 제공해 드리겠습니다. 다만 사전에 등록을 마치시고 착석해 계신 분들 먼저 제공해 드리는 점 다시 한번 말씀드립니다. So we'd like to serve you the meal and we prioritize those who pre-register for this event and seated while we are serving the meal. Thank you for your understanding. 네, 점심 맛있게 드십시오. 네, 여러분 점심 맛있게 하셨습니까? 저희 오후 프로그램 1시 25분부터 오후 프로그램 시작하도록 하겠습니다. We'd like to begin our afternoon program 1:25 sharp, so please return to your seat. So welcome back, everyone. I hope all of you are having recharging lunch break. So we're gonna resume our afternoon program with session 2. Session 2 is titled AI-based strategy for national management of bacterial infectious disease. And to lead this meaningful session, I'm going to introduce you our two co-chair. First of all, Dr. Heer Che. Heer Che is now currently serving as a Director General for Infectious Disease Research at National Institute of Infectious Disease. After earning his PhD at the veterinary medicine at Chungbuk National University, he has been a leading figure in fields of bio big data and biomedicine. And his co-chair, Professor Seung Ki, is holding triple degree from Jeonnam National University. He's wearing double hats for now. And he's leading the Korean Society of Diagnostic Immunology and also heading the Biomedical Research Institute at the Jeonnam National University. So they're gonna lead and moderate the session 2. So please give them a big hand.
예, 방금 소개받은
Hello, good afternoon. I'm Ki Seung-jung as introduced. And I'm Che here from NIID. So in this afternoon session, session 2, we are going to have a discussion about AI-based strategies for national management of bacterial infectious disease. So the Professor Ki will cover first 2 presentations. Remaining 2 presentations will be supported by me. So let me introduce the first speaker. For this session, we are going to have Cesar Dera Yu Pente. He is working at the University of Pennsylvania. So microbiology, immunology is the field of his expertise. And he received a PhD degree in University of Pennsylvania. So there is a recorded presentation material, so we are going to share that. So there will be no Q&A session for this presentation. So let's begin.
For the very kind introduction, and it's great to be here, at least virtually, to tell you some of the work that we've been doing for over a decade now, applying AI methods and computational methods for antibiotic discovery. And so I remember when I was still in my early 20s doing my undergrad, and thinking whether we could find something within biology, if we could find gain some understanding of biology that we could then use to solve some of the greatest problems of our society. And I did this analysis of the biggest problems, and this is a partial list of that. And I found that actually the most underinvested area in the world that affects the most people is actually the problem of antimicrobial resistance. And these are bacterial infections that are increasingly resistant to every antibiotic that we have available. And so trying to find antibiotics became sort of my ultimate mission. And so by studying biology back then, like I mentioned, I was wondering whether there was some sort of entity in biology that we could use to solve all these things. And it was amazing to see, to learn that it actually exists. The minimal functional molecule that exists in life is actually short chains of amino acids that are called peptides. And so I've been fascinated ever since with these peptide molecules and trying to learn how to program them by using many different tools, from chemistry to computer science and so on. Antibiotics, it's a crucial problem. Antibiotic resistance, it affects every corner of the world. And currently, these bacterial infections, they are associated with about 5 million deaths per year, and the projection is that this is going to get a lot worse. It's going to be around 10 million deaths per year in the world by the year 2050. As we can see in this graph, this is projected to surpass every other major cause of death in our society, including even cancer. So it's a huge global health problem. I think, in my opinion, antibiotic-resistant infections are one of the greatest existential threats facing humanity, and really, I've really devoted my whole career to trying to think outside of the box to try to come up with new antimicrobial agents. And like I mentioned, I've always been fascinated with these peptides, peptide molecules. If you analyze nature and biology, they are the simplest nanomachines that can do functional activities in biology, they're an ideal training ground for AI, really, because they have an astronomical sequence space, which would be impossible for humans to sample in the lab. And so algorithms are ideally suited to sort through all of this chemical diversity and chemical space. Peptides are small and experimentally tractable and therefore scalable. And so we actually have robots, chemical synthesis robots in my lab that enable us to make them in a high throughput way. And so then that makes the research a lot easier. These compounds are also, they're very tunable molecules, meaning by modifying the optimization function, you can tune them or optimize them to become antibiotics, which is a lot of what I'll talk about today, but you can also tune them to become anti-cancer agents or immunomodulators that can modulate the immune response. So they're really amazing molecules in that sense. And so these are some of the reasons why we think peptides are the ideal proving ground for really AI research in biology. And I'm going to tell you just what we've done in this field over the last 6+ years. And this all started when we asked this fundamental question of, can we use AI to accelerate antibiotic discovery? And so first I'll tell you how antibiotic discovery is done traditionally. So it's a really physical process where researchers go around nature and they take soil samples and water samples from nature and they try to purify active compounds from all of that complex organic matter. But as you can imagine, this is a process that relies on a lot on trial and error experimentation. It's very unpredictable, and oftentimes it does not yield any good candidates, right? So instead of doing that, we decided to propose a new paradigm for coming up with antibiotics, which is, why not take advantage of the decades worth of biological data that we have at our disposal in the form of genomes and proteomes and metagenomes that have been sequenced over the years? And why not conceptualize biology as an information source of a bunch of code, essentially, that can be explored with the right algorithms to try to find new hidden molecules? So our systems, basically the analogy is that they operate like magnifying glasses that enable us to zoom into biology, the complexity of biology, to try to find new compounds, new molecules that might be of interest. And sort of this is the analogy, right? Like it operates like a barcode that samples through all biological systems and like genomes, proteins, and so on, and is capable of finding new antibiotic agents. So this was the paradigm shift where we sort of conceptualized biology as a bunch of information where we could find new molecules. Another motivation for this paradigm of using AI is that it takes a long time to discover new drugs, including new antibiotics. It can take up to 6 years to come up with some relatively promising candidates, and it's really expensive. And so we thought with AI we could really dramatically sort of hopefully accelerate this process of discovery. So I'm going to tell you chronologically how we went about this. We started by actually taking inspiration from image and speech recognition algorithms. But of course, in our case, we didn't want to recognize faces or sounds, but we wanted to recognize molecular patterns that constituted potential antibiotics. And so that's what we did. We came up with this scoring function algorithms that could sample entire proteins to see if we could find antimicrobial molecules hidden or what we call encrypted within those proteins, right? And a protein, you can visualize it in 3 dimensions like we see on the screen, but you can also visualize it in 2 dimensions with the amino acid code here in the y-axis. And our algorithms, essentially what they do is they run through this code very quickly, and they identify regions within the code that are predicted to be antimicrobial, and these are given by this different coloring scheme, which is all driven algorithmically. So of course this is just theoretical work, right? It's a prediction because it's all directed by this algorithm. But it's a very powerful approach because it enables you to sample entire proteins and within the complexity of that protein, identify regions that are predicted to be antibiotics. For example, this fragment that we see here in yellow. And then we also have synthetic biology tools in my lab that we can utilize to extract that yellow fragment from the protein to try to develop it into something useful. So we did this for a number of proteins. We sampled them, we explored them, but quickly we realized that with advances in compute power, we could actually scale up what we could do, and that's what encouraged us to actually embark on this amazing journey of trying to sample for the first time the human proteome as a source of antibiotics. The human proteome is essentially all the proteins encoded in our genome, so essentially our entire genetic code. And we decided to utilize this scoring function algorithm to see if we could find new antimicrobial molecules there. But this was really a huge scaling challenge because we actually wanted to sample 42,000 proteins. We went beyond the prototypical 20,000 gene-encoded proteins. We also considered isoforms. So again, 42,000, right? And we were used to just doing one at a time. And so this was we had to calculate sort of the scaling law in order for us to achieve this. And it was really remarkable that our algorithm, because it was really computationally inexpensive, it was capable of sampling through every protein in our body in just about 1 hour with the compute power that we had at the time. And so the exploration was completed in just 60 minutes, which was really, really a surprise to us, but it sort of opened new avenues for how we think about the application of computational tools in biology. And this approach, it enabled us to discover, more from a biological perspective, discover thousands of new molecules that were previously unrecognized to have antimicrobial properties in our own body that we call encrypted peptides, because they're encrypted or hidden in many of the proteins in our organism. And then in my lab, we not only do the computational work, we have a computational lab, but we also have a chemistry lab where we synthesize a lot of these peptides with the robots that I mentioned earlier. And so what we did is we synthesized dozens of these computationally identified molecules, and we tested them in the lab. We also have a microbiology/synthetic biology lab where we test everything that we discover in order to validate whether the computer was correct or incorrect. And we got over a 62% hit rate, meaning out of the compounds that we synthesized in the lab that were predicted to be active by the algorithm, about 62% of them were actually active at killing bacteria in the lab, which was really amazing given the simplicity of the algorithm that we use here. And then we tested them. We do all the preclinical work as well in my lab. So we test whether the computationally designed or discovered antibiotics, whether they have safety and efficacy, good safety and efficacy profiles in preclinical mouse models. So that's what we did here, where we developed this skin abscess infection model. Where the mice are infected on their back with bacteria. And a biofilm-like infection is formed of the back of these mice, and then we treat them with in this case, our encrypted peptides that we found in the human proteome. And here are just selected experiments. We actually ran this in 2 different mouse models, but this is just one of them. And these are 2 different experiments where the mice were infected with either Pseudomonas aeruginosa or A. baumannii, 2 drug-resistant gram-negative pathogens. And in gray, we have the untreated control group of mice in each case. So you get a sense of how many bacteria are present in those mice that are left untreated. And then we have a treatment with one of the peptides in red, or the other one here. And then in purple, we have a cocktail, a combination of these 2 compounds in a cocktail. And we can see an even more drastic or dramatic decrease in the infection when we have that cocktail. So this indicates synergistic interactions when you combine them, which was really, really nice to see as well. Out of the thousands of compounds that we found, these are 4 examples where they seem to play a role physiologically. So we can see that they're produced in the body naturally at different levels, from low to medium to high, and in different parts of the body: the cardiovascular system, digestive system, nervous system, and so on. So the current hypothesis that we have is that we believe that these small peptides that we're finding, we believe that they play a role in in host immunity. So they protect ourselves from invading organisms like bacteria by providing a first line of defense to counter them. And so we're proposing that peptide-based immunity perhaps is a lot more significant and substantial than previously thought. And this is an ongoing hypothesis that we're testing in my lab. But this work also sparked many other ideas and many other new hypotheses in my lab, one of which was the fact that we're finding all these new compounds in the human proteome, most likely means that they're not just confined to the human body. And so we came up with this idea that maybe we would find them all throughout evolution and across the tree of life. And that's when we decided to look at our closest ancestors to see if we could find similar compounds encoded in their genetic code. And of course, our closest relatives are Neanderthals and Denisovans. So we decided to investigate them, to look over their biological information, biological data, to see if we could find new antibiotics. And in the process of thinking about all this, we came up with a new framework for identifying molecules and studying molecules, which we call molecular de-extinction, whose ultimate goal is to really identify molecules throughout evolutionary history, to then being able to synthesize them in the lab using chemistry, and then to compare them throughout evolution, like to essentially analyze how certain mutations or changes that occurred over time influence their biological function. And because we work with peptides, which are the minimal functional entities of biology, we can, for the first time, really study how changes that took place through evolution actually influence the biological function. And that can be, of course, antibiotic properties, but it can be also immunomodulatory activities or ability to target cancer cells and so on. So we're really fascinated by this framework of applying it to applying this framework to history, to evolutionary history. And if you think of molecules, they're actually documents of evolution, right? And we believe that they harbor a lot of information that of course, in the ones that were extinct, it got lost through time. And so we think we can now extract a lot of that information to try to inform how we design, in this case, new peptides to solve problems. But going back to archaic humans, so in order to investigate Denisovans and Neanderthals, and in order to see if we could find antibiotics there, we developed this machine learning model that we call Pancleave, that are really high level. What it does is it takes all the proteins from Neanderthals, let's say, it chews them up in little fragments, as we can see here. And then we run two different filters, one of which is a machine learning filter, the other one which is a human expert filter, in order to predict whether those little fragments will be antibiotics or not. And I highlight this, I really underscore this point, because every critical decision that we make in my lab, we take into account the recommendation made to us by the machine, but also the recommendation made by the human scientists, the human experts in my lab. And I think this is pretty predictive of how science is going to go in the future, where I believe it's going to be a really tight collaboration between the power of machines and then human ingenuity. And I think in the loop, and I think that's going to be really essential in how we evolve the scientific, how we apply the scientific method in research, and how science and innovation evolves into the future. So going back to this machine learning model, it enabled us to identify the first therapeutic molecules in extinct organisms. Here we have some examples in Denisovan proteins, where we found were predicted by the model to be antibiotics, and also in Neanderthal proteins. And we use the modern human proteome as a control in every experiment that we did theoretically here. This is all just a prediction. So then, like I mentioned, we have the capability to make all these molecules using chemistry. So we did that. We saw that some of them had activity against pathogens in the lab, and then we tested them in preclinical mouse models, which I'm showing here. On the top, we have the skin abscess infection model, which I described earlier. And on the bottom, we have a deep thigh infection model, where the mice are infected. They have these deep infections in their thighs, and this is the mouse model that really the Food and Drug Administration, the FDA, wants to see for any sort of promising preclinical candidate. And I won't go into all the data, but Neanderthaling was the top hit in these experiments. We also, I haven't mentioned this, but we had to come up with new names for all these compounds, all these molecules that we're finding. So the convention that we have decided upon is that we name them based on the organism that produces them. So of course, Neanderthaling comes from Neanderthals, and you'll see many of these names throughout the presentation. So this was really exciting to us because it demonstrated that we could apply the framework of molecular de-extinction plus machine learning in a practical way to come up with new antibiotics, in this case, that worked in the lab and in a lab setting. And so then this gave us perhaps the courage of asking a more ambitious question, which was, if we're able to do this, why not just try to sample every extinct organism known to science? But similarly to what happened earlier when I told the story of the human proteome, this was a huge scaling problem to us, because here we're just sampling the genetic code of 3 organisms, right? Neanderthals, Denisovans, and modern humans. But now we want to explore every extinct organism. That's hundreds of them. So this required us to develop a more powerful AI model. And so we developed a new AI model that we call Apex, which is, we developed this and we trained it from scratch in my lab. And this is the architecture of Apex. And the really remarkable thing of this model is that you can give it any amino acid sequence, any sort of genetic code, and it's capable of directly predicting the antibiotic activity. So it's a sequence to function prediction model. It doesn't take into account structure. So arguably, it's even a harder problem, right? Because you basically, all we're giving the model is a chain of amino acids, and it's already, the model is predicting what those amino acids, what that functional molecule is doing out in the world, in this case, killing bacteria. And in the future, we believe that it will be possible to devise AI models that are sequenced to function for other problems, including cancer, immunology, neuroscience, and many other things. So really, and really Apex, it really enabled us to sample organisms all throughout evolution, all throughout evolutionary history, like I mentioned, including the past. So it sort of opened a window into the past, and it enabled us to sample biological data from the Holocene and the Pleistocene. Here in this schematic, I'm showing representative organisms where we found the top antibiotic candidates. It includes ancient penguins that were extinct in the 50s, ancient zebras, magnolia trees that disappeared throughout evolution. It includes ancient elephants, woolly mammoths, and even sort of giant sloths that existed, many years ago. And many other creatures from the past, right? So it essentially, for the first time, we're able to sample ancient biology to try to extract and analyze all of that biological data to try to find new useful molecules. To give you a sense of the scale of this mining approach that Apex did, these are all the organisms that we sampled. And as we can see, we have prediction and also active. When we say active here, it means that we actually synthesized those peptides and we validated them in the lab against bacteria in vitro assays. And then we tested them, of course, in preclinical mouse models, which we typically do in my lab. And once again, on the top here, we have the skin abscess infection model, and on the bottom, we have the deep thigh infection model. And sort of to summarize these results, the top candidates that we identified came from the woolly mammoth molecule that we call mammutocin 2, and others that were really promising came from the ancient sea cow, the giant sloth, and the extinct giant elk. And in this case, in some of the compounds that we found, some of the small peptides, they exhibited anti-infective activity which was comparable to the state-of-the-art sort of antibiotics that exist. For example, polymyxin B, it's a conventional antibiotic that is only used as a last resort compound when nothing else works in hospitals to treat an infection. Clinicians use polymyxin B. And here we identified, by looking into ancient biology, we identified molecules that were really comparable to this antibiotic. So this was hugely exciting. This work made the cover of Nature Biomedical Engineering, where the journal highlighted this idea of really this new concept of identifying useful molecules like antibiotics in ancient organisms.
So actually, this video clip was 40 minutes. We are going to share the original video clips later.
Next presentation will be made by Professor Hyungjun Kim from Seoul National University Hospital in Bundang. He is assistant professor and he received bachelor's and a master's degree as well as PhD at the Seoul National University. Now he is professor at the Department of Internal Medicine at this hospital. Thank you for the kind introduction. I am a clinical doctor. I face patients every day. Especially, I face TB patients every day. My presentation is not about complicated content, but my presentation is based on the perspectives of a clinician. And I would like to talk about what patients need, and in order to meet their needs, what kind of research we need to conduct. This is my COI. And this is the content of my presentation. First of all, why do we need AI in TB? And before myself, a lot of machine learning or AI experts make presentation. I would like to briefly give you an overview of machine learning and development of machine learning models in TB and chest X-ray based diagnosis and triage, outcome prediction, and clinical decision support system. Then why are we talking about AI in tuberculosis? It is a communicable disease. It is a communicable disease and it requires a systematic control. And TB often has a high prevalence rate in low and middle income countries. And once infected with TB, there are interim phases before the TB becomes active. And the highest sensitivity comes from culture, but culturing the bacteria takes a long time. And genetic test gives you results quickly, but it is expensive. Then we can use X-rays. It depends on the experience of the physician. So TB requires a systematic control. We need a somewhat uniform and consistent criteria to manage TB in a systematic way. So when we talk about AI, we think of prediction models. There are many AI experts here, but just I would like to share with you how physicians understand AI. First, we do labeling, training, do the validation, and test the model. So when it comes, labeling is the first stage. In the clinical medicine, labeling is very difficult. Outcome of interest, it does not exist in just 0 or 1. And in many cases, labeling is difficult to be done in 0 and 1. For example, the color of the face of the patient, level of extent of cough of patients, subjective symptoms felt by the patient, chest compression of patients. These are the elements we cannot exactly label into 0 or 1. These days, machine learning is known widely, so we use machine learning often instead of labeling, and then we train the model, do the validation, and then test the model. So here we have training set, validation set, and test set. We create a machine learning model and run validation and test the model and get the final score of the model. Clinicians engage in raw data generation, pre-processing, and data labeling processes. For these processes, it takes a long time. And for score, it includes NC value and accuracy value. Model performance is based on AUC value. As you know, if it's higher than 1.0, it's valuable. confusion matrix. It has predictive label and actual label, and it has this kind of table and calculates positive and negative labels. Then I will share with you actual cases. First, this study wanted to diagnose TB pleurisy. There's the criteria of diagnosis for TB pleurisy, but that's not very clear. So it depends on the opinion of the physician. So they wanted to borrow the power of AI. In Shanghai, for 400 patients, they conducted this study. Patients with TB pleurisy, malignant effusion, which means cancer, and parapneumonic effusion. So these patients participated in the retrospective study, and these are the features. The clinician collected features like demographics, symptoms, and laboratory findings, and then they created machine learning model. Prediction, and then they compared the model with the previously well-known variables to see how good the machine learning model is. And demographics include variables like sex, age, history of smoking. It includes lab test results. It also includes the pleural fluid test results. And ADA is what we want to compare. If ADA is higher than 17.5 U per liter, then the accuracy will be 91.6%. That's not bad. But researchers wanted to create a good model. So they conducted logistic regressions and they used random forest method, and random forest model showed the highest performance with the accuracy of 91.6%. So machine learning model became the winner here. More specifically, among machine learning models, they applied various features and variables. What is the most important feature? It is ADA, which is followed by CEA. CEA increases when the patients have malignant effusion, and there are other variables. Then we have a question here. ADA alone gives us sufficient accuracy. Then why do we have to collect all these variables and run the AI model with Python? We have to apply input patient information, but still we cannot get the 100% perfect answer. Between clinical gain and complexity, we have certain trade-off. This is a challenging slide. I'm sorry to show you this slide, but this slide shows some pessimisms. It seems like what machine learning did was just a general statistical analysis. Then what's the special difference? In clinical medicine, the biggest issue of this AI model is that however good a model might be, it is very difficult to actually use the model in our daily clinical practice. For example, we meet patients, one patient for 3 minutes each time when we meet patients, and how can we efficiently use this kind of model for the 3 minute time, for the 3 minutes we meet patients? And AI rapidly grows, and new models or better models appear very quickly. So once we adopt an AI model, soon we see another better model comes up very quickly. And For many patients, it is difficult to take the anti-TB drugs consistently, so many of them just stop taking the medicine. So in Brazil, there was a cohort study for patients with pulmonary TB. They excluded patients who stopped treatment, and they excluded patients who were transferred to other hospitals, and they measured features like these. These features were defined by the researchers. And, and they wanted to see how AI model is good in predicting the loss of follow-up patients. And they ran the machine learning model, but it was not really easy to predict this based on the machine learning model. And they had so many features. So having so many features could make it difficult to have good performance in the model's prediction. So they shortlisted the features like prior TB experience, the smoking experience, sex, and whether the patient received relevant education before. So if you look at the results here, this is not an excellent model, but it is not bad. So the important thing is how are we going to apply this to the clinical practice? On this is the website of this Brazilian research. I entered my information, and the model says I have a 25% probability of stopping taking medication. So I personally also thought that I might try to stop taking medication if I feel it is really cumbersome or it's not easy to take the medicine. So in clinical medicine, it is very important to turn patients into data, into binary information. Patients they are humans, but we have to put the humans into binary information, 0 or 1. If we have too many features, it shows us lower performance. If we have small features, the model gives us lower accuracy. But there's a kind of information about patients that we can make a very good use of, that is X-ray. We can make the complete use of X-ray information. This is a research conducted in Korea. This is a single center. Here, they use the single center data for training a validation set. And for test set, we use the data sets of 6 external institutes. And radiologists review the X-ray image and diagnose whether the patient has TB or not. And we create a machine learning model that calculates the probability of TB based on the image. And this model showed us very excellent AUC of up to 1, and the model's accuracy was also quite high. We compared the performance of AI with physicians. It was better than physician. It was similar to radiologist performance, and performance was similar to that of thoracic radiologist. So here you see a lot of models. The previous slide was about the model we created in Korea, and now this is the case in South Africa. For 770 patients, they conducted TB prevalence survey. They used 12 CAD models, and they looked at the AUC. The AUC was from 6.6 to 0.9. And there's a study about meta-analysis. They used 5 software products based on 21 references. And these were the modalities used. And sensitivity was from 86 to 91%, specificity from 59 to 80%, AUC from 0.8 to 0.9. The lunit here shows quite good AUC level. So the result is good. Then can we really put our trust in this model and can we actually apply this to the clinical practice? In 2024, there was a consensus meeting at WHO. In that meeting, they talked about TB diagnostic pathways in a situation where they don't have enough resources. So WHO came up with a criteria for TB diagnosis in resource-limited setting. So they defined that in terms of the characteristic, the optimal test should have 95%, at least 95% diagnosis sensitivity, and at a minimal level, the sensitivity should be 90%. And the price should be $2 or less, at least. Then what do you think of this? Do you think the price is okay? And there was a simulation-based study. They estimated the cost for AI-based chest X-ray interpretation. In 2021, this study was conducted in Pakistan. By using AI, they interpreted X-ray, which cost $2.7, AFP smear cost $1.2, expert test $21, and radiograph read by a clinical officer 30 cents. If it is read by a doctor, 45 cents. This is the level of cost. So they ran a simulation. This simulation is based on the existing data. In Pakistan, there was a cohort study. If a patient visits a physician and if the patient has TB symptoms, the physician could have the patient receive a sputum test or have the patient data entered into the AI system. So first, in the simulation, patients without HIV and with TB symptoms were they participated in this testing. And if you look at the cost, if smear is the basis for microbiologic test, the cost is $1,756 per 1,000 persons. If you use AI, the cost will drop down to $747. And if you use expert system, the cost will be $19,000 per 1,000 persons. If you use AI-based chest X-ray interpretation, the cost goes down to about $5,000. So if there are patients for whom TB screening is required, and you can use the the WHO recommended rapid diagnostic tests. And WHO came up with another policy statement with somewhat softened standard. For example, the standard for specificity and sensitivity became softened before, and six CAT software products met this newly defined standards of WHO, including the software made in Korea. So these softwares were recognized by WHO. So we talked about whether the system AI or machine learning system is good in terms of the diagnosis of TB. So we also need to look at the level of TB in patients. So we created a machine learning model. For training and validation, we used a multi-center retrospective cohort study. For tests, we had two sets, and we divided this into the period before the diagnosis of TB and after diagnosis of TB. The performance is here. As for the X-ray score,
베리어블들하고도 어느 정도 상관성이
they
있거든요.
have correlation with the
특정 스미어,
severity of TB.
아예 근육 많이 나오면 스미어 포지티브가 많이 되겠죠. 그래서
So if there are a lot of TB pathogens, then the smear test will show more positivity, and the score will decrease with anti-TB treatment.
그러면 이제 이걸 어떻게 활용할 수 있을까요?
Then how can we use these
저는 이제
study
영상을
outcomes?
찾아봤는데 6개 우리나라 병원에서의
So in Korea, 6
연구였고요.
hospitals conducted retrospective study,
그다음에 엑스퍼트 값이 있고
and the study was about adults with rifampin susceptible pulmonary TB, and the number of patients here in this study were 200 patients. 230. Among 230, 200 patients successfully received the treatment and were cured, and 24 people, they died. And regarding treatment success, there are the factors like people with younger age, females, those with higher BMI, those without hematologic malignancy, and those with lower TB extent scores showed better outcomes in their treatment. And at 8 weeks after the treatment, if their culture converts from positive to negative, they had higher probability of successful treatment. And lower the initial TB extent score, the better their treatment outcomes. So 6-month HRE, in terms of 6-month HRE, TB patients show similar trends, but if you look at each patient, the life is not really easy. They have to take 10 tablets of drugs a day, and they often experience adverse events as well. And they go through a lot of uncomfortable challenges in their lives while receiving the treatment. Now, I'd like to talk about clinical decision support system. It is a kind of algorithms. When physicians make a decision, they can use this system. So we have two types, knowledge-driven CDSS and data-driven CDSS. Knowledge-driven one gives you a guideline based on the expert's opinion, and data-driven CDSS has machine learning combined, and it has some algorithms. Each patient has different pathogens from each other. So what kind of TB treatment do we have to use? When you make a decision about this, you can receive help from CDSS system. You can refer to the knowledges of other physicians when you make this kind of decision, when you use knowledge-based CDSS. First, you set your goal and meet with experts and create a prototype. For example, when we have this kind of patient, the treatment should be done in this way, for example, and then you run the machine learning model. So for different type of patients, you can suggest different types of therapeutics or treatments. So in this way, the CDSS system comes up with the suitable treatments or therapeutics for different patients, and the outcome you get from the CDS system, you need to review whether the results from the CDSS is proper or not. So when I ask TB experts about something, for example, if I ask 10 TB experts about a certain aspect, they give me different answers. And there are various drug-resistant profiles, for example, 129 different drug resistance profiles. And there there are about 10,000 treatment regimens. So when experts come together, they can create a prototype about how they're going to give treatment to the patients and refer to the information here. They run the machine learning model and get the final score. If there's a certain TB patient who is resistant to a certain drug, then the machine learning gives you recommendation about adequate drug for the patient. So if the system recommends you the adequate drug for the patient, you can say yes or no, depending on whether you agree with the answer. So here, it asks you, do you agree with the regimen number 1 recommended from the system? If you look at the result here, 78% of the experts said yes, and the others said no. So experts or physicians have different opinions over the proposals made by the system. So what's important is that you need to ensure the good quality in the questions you ask. So you have to define clinical questions very well and input high quality labeled data. And X-ray based AI tool showed so far the good potential for TB screening and disease activity assessment. And we need to conduct more study on CDSS model in terms of their clinical decision supporting function. And ultimately, we have to think about how we can apply this system to the treatment of patients. Thank you very much for your attention. Please raise your hand if you have a question.
Wonderful presentation. I am Pravin. I'm from the International Vaccine Institute. My question was regarding the PFCA levels in TB prediction. So what is the significance that PFCA levels were also as high, almost as high as the ADA levels? So I couldn't understand what high PFCA levels mean in this context. Thank you so much.
Yeah, thank you for the great question. So let me make it straight. You mean why does plural CEA levels matter in the model, right? Yeah, it's because of the patient group. They included TB pleurisy and also malignant effusion. So if the patient had malignant effusion, the plural CEA levels would be high. And if the patient had TB pleurisy, the levels would be low. So that would discriminate between TB pleurisy and malignant effusion. So that's why it number 2 in the model.
예, 또 다른 질문 있으시면 해주시고.
Another question? Now, let me ask you a question. TB is one of our tasks. For patients with active TB, we're trying to develop global biomarker. We excluded the cheapest chest X-ray. Seems that chest X-ray has good potential. Currently, we are using expert system, and AFB smear and culture have become the gold standard. And as for expert system, we cannot use it for follow-up. It is used for early diagnosis. When we do follow-up, we have continuous the DNA issue. There's microbacterial load assay. If you do this based on RNA, you can do some treatment. So the relevant technique was developed in the UK. We have not yet adopted this. And in Japan,
MPT 64 엔티젠에 대한 객강에서의 항원 그 정량 검사가 있거든요.
there's a the assay related to the sputum. And is there a clear difference in terms of the activity level if you compare this with the chest X-ray? So what you're doing regarding TB is very important. TB takes place a lot in low or middle income countries, so we cannot ignore the cost element. X-ray is not perfect, of course. So once patient gets TB, it gives them the side effects, or it gives them the traces of the disease. So when you develop biomarkers, so considering the cost issues among TB patients, I think it is very important to develop biomarkers. Any other questions? I cannot force you to ask a question. This will be all for this presentation. Thank you.
So before moving on to the later second part of the session 2, we'd like to take a 10-minute break. So we're going to resume 2:35. Thank you. 네, 여러분 잠깐 10분만 쉬었다가 이어가도록 하겠습니다. 10분 후 2시 35분에 다시 시작하겠습니다. 잠시 후 뵙겠습니다. So time's up, everyone. So this is a high time to resume for the remaining session 2. So just like before, I'd like to give my floor to Dr. Che and Dr. Gi. So please give them another applause. 여러분 큰 박수로 이제 다시 좌장을 맡아주신 두 분께 응원 부탁드리겠습니다.
네, 오후 두 번.
So we had two presentation after lunch, and so we have two remaining presentation. So today's discussion is about AI. So during the break, and actually we had some discussions. So we can collect data from AI. So I use a Gemini. So if there is a wrong information, please correct me. So we are going to invite William Shao. So, well, according to my search, and he has expertise about biology, and as for the infectious disease and the study, he has a great understanding, and actually he has a One Health approach, and so he is a world-renowned researcher. That's what Gemini said. So today, and well, he's going to share information about the Salmonella. So the outbreak cluster related study can be shared. So what our accessory genome tells us is the title. Please welcome him with a big hands.
Thank you very much for the invitation and for the warm welcome. So I'm William Shell from Canada, and I'm going to tell you a story about a study that's done by a bright student of mine, but then that would lead to a general message about why it is important to work together and to collaborate. So after sampling all the amazing food that I had in Korea, I guess it's only appropriate to talk about food safety and why it is important to safeguard our food sources and to make sure that we don't get food poison. But foodborne diseases is a commonplace, and on average, 1 in 8 people around the world, this is really talking about the more developed regions suffer from foodborne illness every year. So, for example, 1 in 4 of Americans, 1 in 6 in Canada, and the 1 in 8, I guess, is sort of the global average. And the number of hospitalization and death due to consumption of contaminated food in U.S. has doubled in 2023 and to 2024. Food recall has also increased by 8% in the U.S., and diseases such as salmonellosis, listeriosis, and disease caused by E. coli have also increased by 41% this is all U.S. data. And Canada is actually not that much better. So if we look at the food safety investigations by Hazard Canada between 2020 and 2025, you can see reliably that investigations that are of microbiological agent-related cases, as shown in purple here, has increased year over year as well, suggesting that we need a better approach in detecting foodborne pathogens and also tracking these pathogens better. So because food is a globally traded commodity, collaborative networks that are global in nature is needed to serve as the monitoring agencies for shared pathogen information. And this would then enable, for example, timely outbreak alerts, traceback investigations, disease burden estimations, and information on how to improve the policies, and also assess the intervention effectiveness. For example, vaccines for poultry stocks or other types of interventions, are they actually reducing the disease burden or just provide another evolutionary pressures for the pathogens to shift into a different variant? So the most well-known example is PulseNet International that has global coverage in terms of tracking foodborne pathogens. So I'm going to use foodborne pathogens as an example, but the approach that we're taking can actually be applied to hospital-acquired infections and other types of infectious diseases. And genomics data serve as a powerful tool of linking cases. So we know that a lot of foodborne outbreaks have a common source or common exposure. That's often due to a rapid expansion of a specific strain or specific type of organism. So as a result of that, a rapid spread of this pathogen then manifests into clusters of cases that are colonized by clones or by characteristically similar pathogens. So therefore, the clonal structure of the pathogen due to clonal expansion therefore allow us to use the molecular data, including genomics data, to track these cases. However, we all know that these pathogens are extremely diverse. So today I'm going to use Salmonella as an example, partly because it's a leading cause of foodborne illness worldwide. It's an important pathogen. But another reason is that compared to E. coli and some other foodborne pathogens, it actually has a relatively stable core genome. And it's, so for example, looking at these Salmonella enterica pangenome distribution, you can see that the core genome consists of about 2,800 genes, but each of these serotypes have additional serotype-specific genes that are not found across all serotypes. So the strain or lineage-specific genes, what we sometimes call accessory genes, are often associated with mobile genetic elements. And the question is, can we use these in addition to the core genome to improve the tracking of these pathogens, given the known diversity? So common genome typing methods that are used in public health often just focus on the core genome. So these include the traditional MLST, which look at just a handful of loci, handful of genes. But also, even if we enter the genomics era and looking at the core genome MLST or reference-based core genome SNP analysis, they're mainly focused on shared genomic regions across these strains. So we would like to understand what type of signals and what kind of additional information can we get from the accessory regions it provide. And as shown in the scenarios down here, oops. If you look at only the coding genes as shown in these blocks, then if there are intergenic variations as shown here, or if you're looking at reference-based approach where, again, you're looking at the genes, the shared genes, and ignore the accessory genomes, then you can see that there's a lot of information that's left out in your analysis that can potentially be informative. So the key questions that we wanted to answer were what genomic features are predictive of Salmonella outbreak clusters? Why are these genomic features predictive? And can these predictive features actually be transferred to new outbreaks, new clusters that we have not seen before? And the data sources was actually painstakingly curated manually from reading papers. So Jimmy, who's a student, manually curated 65 different outbreaks from PubMed and also from the NCBI pathogen detection portal. So we divided the 65 outbreaks into a training data set and a validation data set. For the training data set, there's about 200 genomes from 24 different outbreaks, and they represent different serovars of Salmonella. And each of the clusters have 5 or more isolates within each of these outbreaks. And the validated data set is smaller, has 104 genomes from 34 outbreaks. And you can see here that it's not quite comparable, that it has, on average, a lower number of isolates per cluster. But it still has different serotypes represented. And this is potentially an issue that we only caught after the fact, after the paper was done, the analysis was done. And this is the paper, the work was published last year in MBio, so the reference is down here. So this is the overview of our analysis workflow. So we collected the isolates from these databases, and we construct what's called a de Bruijn graph, which is a graphic representation of all these genomes. And we and they're divided into segments, and each segment essentially is separate by a variation. So if you encounter a mutation in one strain that's not found in another, then that segment is broken down, based on where the mutation is. And so if a segment is found in all of the genomes, then so there's no variations across all the genomes, then in other words, it's the core, right? Then we don't analyze that, we remove that from the study, and we only focus on what's called the variant unitics. Unitics is just a different name for these segments. And then we use elastic net regression to build a classifier that essentially select for features that are most predictive of the outbreak clusters. So the labeled outcome is essentially the outbreak identifiers, which outbreak does a particular strain belongs to. Okay, and in parallel, we would then annotate the genome and get the functional annotations of all of the genes and of the other genetic elements. And then we take the predicted list of features that are associated with ability to predict outbreak clusters and look at the results and look at what genetic features are overrepresented in these feature set. Okay, so all in all, we came up with about 5,000 predictive features. Each of these correspond to one of the segments in the genome. And the majority of them map to coding regions, which is not surprising given that, as we know, bacterial genomes, 80+% of the chromosome is coding. But there's also about 600 features that are mapped to non-coding regions. And also among these, among those that are situated in the coding regions, 40% of these are situated in non-mobile genetic elements. So, and also several outbreak predictors, these features map to intergenic and repetitive regions. So this gives us a sense that indeed, outside the core genomes and outside of the coding genes, there are signals that can be useful in predicting clusters or cluster membership more specifically. So then how do we then explain how these DNA segments help to discriminate between the clusters? So what we did here is we looked at one specific, one specific set of data consists of 3 different outbreaks, all from the same regions of Canada and from Quebec. And they're spread over 3 years. So you can see they're labeled by the 3 different colors. And in each of these, except the last one, there's a mixture of food and human isolates. So, and these are epidemiologically investigated cases. And then we looked at 4 different mobile genetic elements: phage, plasmids, genomic islands, which are potential mobile genetic elements that have been integrated into the chromosome and may or may not lose its mobility already, and also CRISPR arrays. And specifically look at the single nucleotide variants and gain or loss of entire plasmids, and again, single nucleotide or indels within the genomic islands, and also the copy number variations within the CRISPR regions. And these features are mapped on the right-hand side of the tree. By the way, the tree is generated based on the core genome. So this is the clustering patterns, right, based on the core genome. And as you can see here, based on the core genome, you can't really tell, if you ignore the colors, you see a straight line, so you can't really tell that these isolates are from 2 different outbreaks, 2 different years. But then if you look at the mobile genetic elements, you can see that indeed they are different. So it shows us that, you know, distinct features can be used to explain and add resolution to the clustering patterns. So it's not possible to take these DNA segments or the unitics from one study to another, because the different genomes may have different sequences. So we came up with a different way of evaluating the approach in a different set of data sets. So instead of just taking the classifier that's trained on one set of genomes and apply to another set of genomes, again, we're training these across serotypes, so there's a very diverse set of sequences. What we end up doing is we built new de Bruijn graphs, and we extract, because from previous slides I've shown that these mobile genetic elements do have signals that can differentiate outbreaks. So then we extract the regions corresponding to these mobile genetic elements and construct subgraphs, essentially just using the presence or absence of these of the unitics corresponding to the mobile genetic elements, and generate a dendrogram as shown here. So this is just a distance-based tree that will show you the which of the isolates share more of these features. And again, each of these subgraphs correspond to one type of mobile genetic elements. So using the annotation subgraph, then we generate the dendrogram, as I showed, from these 4 types of mobile genetic elements again. And we then look at we then compare the trees that are generated, or the dendrograms that are generated from these features against the core genome, or against the core genome variations, and I'll show that. And in order for us to then measure the success or failure of the clustering, we use what's called a monophyletic rate. Essentially, you can think of this as the frequency of the strains that are from the same outbreak end up in the same clade. So in other words, the tree generated from the subset of features is congruent with the epidemiological evidence, right? So the outbreak IDs. Okay, so here's the result, and essentially what we found is that using this approach, the pangenome graph allow us to overcome the issue of having to pick a single reference as a starting point, because essentially it becomes more of a all against all comparison. So this allows us then to interrogate a wide spectrum of genetic events that are found across a diverse bacterial population, right? So for example, in this case, across the different serotypes of Salmonella. But perhaps not surprising, because we already know that if you just look at the mobile genetic elements alone, it doesn't predict outbreak clustering very well. And this is shown here, right? If you only look at CRISPR's plasmid, presence, absence of plasmids or phages and so on, then it doesn't use that information alone. It doesn't group the isolates from the same outbreak together. So the monophyletic rate is low. And then if you look at, so the first 4 is the 4 different types. This one is the combination of the 4 different types. It gives a slightly better performance. But if you look at the more traditional core genome MLST or the whole genome MLST, it gives a better performance than any of these mobile genetic elements on their own. And then if you look at detailed core genome variations using this program called ggColler, the performance is even better than the MLST-based approach. But what's quite interesting is that when we compare the core genome, detailed core genome variation with the mobile genetic information, then the performance is better than the MLST-based approach. And this is also true for the validation data sets. We didn't dig too deeply why the validation data set on average performed better than the training data set, but that might have to do with because the cluster size are smaller and so on. Essentially this is part of the feature study. We also want to expand this to other types of pathogens, especially for E. coli and other pathogens that have high rate of horizontal gene transfer and a much less stable genome, to see if these type of approach also holds for those type of pathogens. Okay, so as I mentioned, data curation was the rate limiting step, because the training and the validation data set all require an outbreak cluster ID as a label. And while many outbreak studies have been published, if you go into the NCBI database and try to extract these genomes, they don't come with the epidemiological information. They don't give you the outbreak IDs associated with them. So it was, again, a long process to curate the data set. And this is especially even more challenged when it comes to trying to collect data set from One Health perspective, because fewer studies are across sectors, and also the types of information that are curated in these studies are inconsistent. So generally, the missing and low quality metadata is a common problem associated with trying to do machine learning studies like this. And there's a salient example from the Gazette database, which is the global SARS-CoV-2 and influenza genomic database. And there's a study published in 2022 showing that, for example, 63% of the sequences are missing the demographic information. So in other words, you don't have the host information associated with the sequences. And 84% are missing the sampling strategy. So you can't carry out robust epidemiological studies because you don't know how the samples are, how the studies are set up. And also 95% of the data are missing patient-level clinical information. So again, these are missing from the databases. They may be available in papers, but then there's a gap between extracting the information from human-readable format to machine-readable format. So this is sort of a switching gear a little bit, is that's why we recognized this issue more than 10 years ago and realized that contextual data is critical for interpretation of sequence data, especially when the microbial genomes are so diverse. So contextual information really is just information about the sequences. So it could be the sample metadata, it could be clinical laboratory epidemiological metadata, and also the methods and the provenance information associated with how the data set is generated and who generated them and so on. And these are important information for a list of activities that I listed there, but won't go through it one by one. So then I somehow managed to convince half of my group, about 10 people from my group, to manually look at thousands of papers, and we end up looking and scanning about 500 of these papers, so related to genomic epidemiology, both reports of outbreaks and also reports of surveillance efforts, to identify common data types that are being reported in papers. So in general, most of the papers, not surprising because they're as part of the requirement for publication, report information on study designs, sample collections, host information, sequencing information. But they actually, when we look at the genetic information, such as mobile genetic elements, antimicrobial resistance, and other detailed epidemiological information, there less than half the papers can report these type of information or annotate this type of information. So the recognition of another gap, right? So you have a gap between database to the papers, but also within the papers themselves, the genetic informations are not always consistently made available as well, or even have been analyzed. So over the last decades or so, one effort in my group is to create data standards and data specifications for genomic epidemiology. We started with bacterial pathogens, but during the pandemic, then we led a global effort through the PHA for GE. So this is Public Health Alliance for Genomic Epidemiology, Gates Foundation funded organization out of South Africa, to co-develop international standard for reporting SARS-CoV-2 genomic epidemiology studies. And through our previous effort and this effort, we came up with essentially a systematic approach where we looked at core elements that are being shared across different pathogens, different studies, and different type of environment and so on, and take a modular approach. So we group the metadata types into different thematic modules. This allows us then to reuse the modules across different applications. So we, as I mentioned, started with really AMR in foodborne pathogens, then moved to SARS-CoV-2, and then subsequently in Mpox, and also more recently in wastewater and high-path AI. And the process of putting the standards into practice is very labor-intensive. It's led by a research associate in my group, Emma Griffiths. And this really involves consulting the users, providing the tools to allow them to carry out curation, the supporting documents, the SOPs, and so on to use these tools. And then there's also the maintenance phase of once your standards are developed, if there's a new terms being requested, you have to make that available to the community as well. So overall, this is a community building effort. You can think of as a process of developing a common language to describe genomic epidemiology studies. And so our effort is called a genomic epidemiology ontology. So this all don't have the time to get into ontology, but the idea is that these standards are not just for humans because of AI, because of machine learning. We want to make sure the standards are machine readable and actionable. So at the back end of a human readable format is a machine readable specification as well. So to summarize, AI analysis benefits from rich annotated data. So both the contextual data and also the predicted genetic features are useful. And the variations from both core and accessory genomes do provide useful signals for clustering outbreak strains. And only with rich and accessible genomic epidemiological data can we really continue to improve the use of AI in pathogen genomic studies. In other words, I believe the challenge for us at the moment is not AI methodology improvement, but really a data cliff issue of how can we get human readable data to machine readable format. And actually one of the tools that we've been experimenting and others too is to use large language models to try to map between free text to ontological terms. And actually we have some success showing that it performs better than a rule-based natural language processing tool that we developed previously. So moving away from sort of expecting all sequence data to be deposited in open and public repositories that comes with minimum contextual information to a system where we can actually have sensitive informations stored more in a locally accessible manner, but that can be discovered by other users is needed. So in other words, our current norm of depositing sequence data with virtually no metadata generate a lot of orphan data sets are not really useful for the community. So the focus for us has been trying to build these interconnected, interoperable data repositories with standardized vocabularies and specifications, and also more flexible data governance models that can then respect the data sovereignty and the data sharing needs across a wide range of scenarios. So encourage people to generate standardized data and then to share it with others, but in a controlled manner rather than just force it to be dumped into an open repository. So this is a challenge, of course, and given the current global conflicts and global climate, this perhaps is swimming against the stream. But again, as I mentioned, I've been thinking about this for 10 years. So my call here is, regardless of the global conflicts and global issues, we should still find a way to work together so we don't spend a lot of money generate sequence data, but really have no way of leveraging them for AI applications. Yeah, so creating a pathway to allow sensitive information and identifiable information to go from private repositories to become de-identified information, and over time less sensitive information to end up in the public repository is something we can think about how we can achieve. And I have a large group of people to thank, larger group of funders as well to thank, but I want to point out work is done by Jimmy and Ivan, who's a software developer in the group. And also the ontology and the data standard work is led by Emma Griffiths and Damien Dooley, who's an ontologist and also a knowledge engineer in the group, and a number of data curators in my group. And again, thank you very much for your attention.
Well, thank you so much for your presentation. And actually, as for the pathogen, so actually data quality matters. So you provide great the suggestions indeed. So time is limited, and we are going to accommodate only one question. So if you have any questions, please do raise questions now.
great talks.
Thank you.
I have actually two questions. I'm sorry. So the first one is you presented 4 different type of the factors like genome RNA and phages and plasmid and CRISPRs. So is it enough for differentiate outbreak strain from the another whole hen genomes? And second question is, how can you integrate your research into clinical side, like in hospital?
Yeah, so the first question, the reason we picked those 4 is just because we have bioinformatic tools that can be used to predict these genetic features. But then, yes, incorporating additional type of elements such as transposons or these integrases, so even genetic elements, and then also integration of insertion sites for these elements and so on could also be useful as signals to differentiate recent horizontal gene transfer events from more ancient ones. So yes, additional features I believe will be useful. The second question of integrating into a clinical use, that is work in progress in the sense that, of course, introducing genomic or sequencing NGS into a clinical setting is still not a commonplace. But more important, even if you have the ability to generate the sequences, to have the automated workflows to create reports and so on, are also just being has been worked on in the last few years, but nothing really solid that's in place. There are companies that provide tools that, of course, allow you to generate report based on whole genome data, for example, virulence factor information or AMR, predicted AMR information. But often, as these pathogens evolve, then updating these tools become a challenge. So again, that's why a collaborative environment where there can be a way for people to share data and to share new variants would be an important will be important in order for the clinical application to continue to be kept up to date with the organisms evolved. But we also have been working with other clinical standard bodies to actually try to define what are the report or useful informations that in a clinical setting. So I'm happy to chat more about that later as well. Thank you. Thank you.
네, 감사합니다.
So the last speaker is from Yonsei University, Professor Min-hyuk Choi. He received the 3 degrees from Yonsei University School of Medicine. Now he is a professor. I asked GPT about him, and GPT said his research is a solution to combining data science with the modern medicine's challenges. Today, he will share with us how he's developing biomarkers for AI-driven by using AI-driven analytics. Thank you for the kind introduction. Actually, my original presentation title included the term of microbiome, but I changed the title, AI-driven analytics for biomarker development. Microbiome appears in the 3 slides in the later in the other part of my slides. First, I will talk about urinary tract infection diagnosis, how I used AI model for predicting UTI. Secondly, I will talk about how we can recommend antibiotics related to blood resection. Recently, LLM, including ChatGPT and Gemini, can be applied to antibiotic recommendation. And thirdly, I will talk about AI-driven microbiome analytics. First, let me briefly explain about myself. So Professor Kim said he's a clinician. That was very impressive. I am also a clinician facing patients. While doing the research, I often forget what I do. I am a physician working at the diagnosis medicine department, specializing in clinical microbiology. In the clinical microbiology lab, we deal with this culture a lot. We culture bacteria, and if we find meaningful pathogen, we conduct antimicrobial susceptibility test to see susceptibility of antibodies, and we share this information with the primary the doctor, and the doctor refers to our opinion and decides which antibiotics he's going to use. So all of my researches begin from this point. First, I will share with you the UTI prediction model we developed. On the left-hand side, you will see circle diagram. From It contains 80,000 patients who visited our hospital from 2011 to 2021 for UTI. Dark blue color in the middle refers to E. coli. Most of them were infected with E. coli, and the others were infected with Candida species. As time passes, the amount of patients with Candida species increased. The number of patients with E. coli gradually decreased. So why is there such a change? We analyzed the reason, and we found that the average age of patients increased. So many of the patients had urinary catheters. Then So we thought there was a change in the urinary pathogen, but there was an issue. When we treat UTI, we culture the bacteria, and until we get the outcome, it takes 2 to 3 days. You might have done this when you receive health checkup. If you do dipstick here, it immediately shows you the change of color, so you can immediately see if you have UTI or not. This kind of quick diagnosis is possible, so based on this, we make the decision to treat the patient. 2 to 3 days later, if you get the result from the urine culture, you can change your treatment method. But urinalysis Urinalysis is based on the situation where 60 to 80% of the patients are infected with E. coli, just like in the past. So if you go to the upper side to the right side, the performance is better. The red the different colors shows which bacteria you're infected with. For E. coli, the performance is very good. Compared to culture, the urinalysis shows very good performance. However, if the pathogen is other types of pathogens besides E. coli, then urinalysis shows us reduced performance. This is related to the aging population. With aging population, if the UTI causing pathogen changes, then it's difficult for us to continue to use urinalysis. So we adopted machine learning model. As mentioned before, due to aging population and the use of polycatheter, we saw the change. Then we could include the use of polycatheter and the aging population in better predicting the UTI, better detecting UTI patients. So we reflected these elements in our model. And in the existing urinalysis, microscopic bacteria existence was considered important, and age and sex information was included. But our model showed more than 10 percentage point improvement in its performance compared to the existing urinalysis. And Professor Hyungjun Kim pointed out a very important thing. The important thing is that it will be good if we publish articles like this after creating a model, but what's important is how can we apply this model to our clinical practice. The doctors are very busy caring for the patients, so it is difficult to adopt the system into the clinical practice. So we used SCL. If you ask SCL for automated urinalysis, you can get the results estimated by AI model along with automated urinalysis. For example, you can see the results like this. This patient has a certain probability of UTI, so you need to conduct this or that kind of test. And at SCL, this service is being done free of charge, but if it doesn't have commercial value, it will not be adopted by other hospitals. And this year, a project named ASC Sprint was announced, and this is a joint project with SCL. And we're going to conduct a clinical trial under the permission of the Korea Food and Drug Agency. So let me summarize this part. Due to aging population, the UTI causing pathogen is changing. It's not just about a change in the pathogen, but it actually reduced the performance of the existing urinalysis. This is why we need to adopt machine learning model to achieve superior performance. And we will conduct clinical trials under the permission of the Korea FDA to apply this efficiently to the clinical practice. And next, I would like to talk about LLM model, including ChatGPT and Gemini. So previous speaker talked about X-ray. Specific research groups or companies create good models. They wanted to create good models to enhance the prediction. These days, if I ask ChatGPT to read and interpret X-ray image, it gives me quite good interpretation. Now, many engineers are developing LLMs, and there's fierce competition among LLM companies like ChatGPT, OpenAI, AI, and Google. So we can ask a broad range of questions to the LLM model. And going forward in the future, the LLM could give us better performance in terms of helping us predicting and detecting this disease. But I come to think about whether it will really help the researchers and physicians in the future. As for machine learning or deep learning, they are very good at structured data. LLM model enables, they received multimodal trainings, and LLM is based on pre-trained data, pre-trained models. So I thought about training LLM with the model I created. Then LLM will have its own existing knowledge plus the knowledge it gains from my model, and then this LLM is going to have better performance. This is what I assumed. In 2024, in Nature Biomedical Engineering, Chen Group from Stanford University announced this machine learning related paper. It is about analyzing single cell RNA. They said they used the embedding technology of ChatGPT, and they trained this data to GPT and ordered GPT to interpret the data. And the GPT's performance in interpreting what they asked was not really bad. So I adopted this idea. before, in Sinchon and Gangnam Severance Hospital, I adopted this idea for patients with lower blood streams. First, I collected data of 32,000 patients. In this way, based on Excel spreadsheet, the date of visit at the hospital, outcome of the patient's prognosis, and the kind of pathogens they were infected with. But LLM reads texts and it changed this information into a case report. At the server, it creates a certain format. It shows age 30, she was admitted to hospital at this point of time, and she was infected with this pathogen and received a certain treatment. And I use the expression embedding. This is what it means to do embedding. LLM does not understand the human language itself, but it reads these computer languages. So the case report you saw in the previous slide was converted to this embedding. If you train LLM with this embedding, LLM better understand this type of embedding information. So 32,000 patients' data was entered into the system like this. Based on this, we had this model make prediction. There's an interesting fact. I just entered numbers and figures, and this LLM came up with text like this patient is relatively young, 37 years old. He was admitted to ICU. He doesn't have underlying disease, so he has a high probability of survival. If I just enter certain albumin figure, then LLM uses its pre-trained knowledge and data and gives me an opinion like this. It sounds like the opinion of a human doctor. So through unsupervised clustering, we cluster the patient's phenotypes. On the left-hand side, this is based on LLM. On the right-hand side, we used PCA model. This attribute scan model. Clustering was very clearly done. If you look at the left-hand side, depending on the prognosis of the patients, the worse the prognosis, the color is red, and the better the prognosis, the color is light violet. If you look at cluster 5, this is infection 100% with a certain pathogen. So if you look at the LLM-based AI outcome, it clusters the phenotypes of patients in just like the way physicians do. And on the right-hand side, you can see the clustering of phenotypes. And I adopted a model for patients with bloodstream reduction. When we investigate the risky factors of a certain infection, we often say that people with underlying diseases, people with cancer, have a higher risk of death, mortality. So, acetaminophen takes place a lot at ICU. Ventilate killer patients comes first with pneumonia, and then the problem gets worse and get infection in the blood. In the morning, we learned about network analysis. So we did the network analysis, and E. coli was closer to women without underlying disease, and they had good antibiotic susceptibility. Azabaito was related to the ICU admission. It was also related to pneumonia and underlying disease. Hercules negative Staphylococcus was prevalent among patients with blood cancer or patients with dementia. So this is the pattern we saw, similar to the pattern we saw from LLM. So the pathogen was a very important factor in the cluster. We can see here the red colors gathered together in the similar location. For blood infection, it is very important factor determining the probability of determining the mortality. This one refers to inpatient death, in hospital death.
30일 사망률은 가장 낮은 A 병원 내 사망률은 가장 높은 이상한 분이
So, but if you look at this cluster, there's a very strange cluster in terms of the mortality rate, in hospital mortality. And regarding this group, we conducted deeper analysis and used fine-grained model to see the cumulative incidence of in-hospital death. Until day 30, the death increased rapidly, and then the trend stayed still. However, in terms of so we saw some characteristics of the patients. Patients with chronic diseases, patients with infection with fungi had higher probability of mortality. So by using LLM, we can find a cluster of patients that is hardly seen in the conventional prediction methods. So if you conduct embedding, you can gain new insights. So LLM-based AI could open a new pathway in your clinical activities. So we are preparing pipeline this. We have machine learning model, deep learning model, and LLM-based model. So we decided to combine all of them together. So we screen machine learning model, and CAMPOST and XGBoost model were included here, and TAP MEA model is here. So we train LLM model with the data from these models and have it predict the prognosis of patients, and also we are trying to create a model that gives us recommendation about what kind of antibiotics we have to use for treatment of the patients. For the last part, let me go to the conclusion first. Antibiotics is a double-edged sword. We use antibiotics to treat patients. It also kills benign benign microbiomes. And many patients reach a stage where they have dysbiosis, and if time passes further, they come to have enteroconal and then the patient is likely to have VRE colonization, and if time passes further, the patients the condition leads to true infection and finally death. At ICU, enterofocus was regarded the most important factor in leukemia, acute leukemia as well. Enterococcus was considered the most important feature. With machine learning, I analyzed this. Enterofocus here is also cited as the most important feature. We compare the data between patients and normal people, and many people think that this is a healthy germ or a healthy cell. It is not really detected among healthy people, but if you use antibiotics more, you can see an increase in the amount of enterococcus, which is likely to increase the mortality of patients. So using broad spectrum antibiotics has risks. It poses risk in terms of the mortality of patients. This is why we need to use antibiotics until appropriate point of time. Then the issue is when exactly do we have to reduce the amount of antibiotics during our treatment of patients? So to answer this question, I decided to develop biomarkers. And here we saw various changes in the pathogens based on machine learning, Candida albicans or Enterococcus look like very important taxons. And if you conduct functional analysis, various pathways are detected. I have some more time, so I want to talk more deeply about this. If you use antibiotics, the beneficial germs, butylate producing germs reduce first. What they do is to produce ventilate, and there's forfeiting network germs, including lactobacillus. And lactate producer or lactate utilizers, they have nothing to eat if vitalate production decreases. So they are affected by the use of antibiotics. So beneficial germs and beneficial microbiomes, they die with the use of antibiotics. And then we see an increase in the number of enterococcus. And also, we see an increase in the number of microbiomes that uses adenine as alternative pathway. If they increase further, the patient is likely to have dysbiosis, and they have many resistant genes. So the patient is likely to have more resistant microbiomes. And then the tight junction becomes weaker. So negative, bad microbiomes are created through the use of antibiotics. That leads to blood infection. So at hospital, E. coli is often observed. We see a lot of E. coli among patients with blood infection. Thank you for your attention. Thank you for your presentation. Due to time constraint, we can take only one question from the floor. Two persons raised their questions, but yeah, we will take two questions. Thank you for your presentation. Regarding your approach to LLM, it was very interesting. My presentation is also related to that area. If you enter the in-house information to LLM model, you might feel some burden because that's in-house information, and we have to advance the features and fine-tune the LLM model. It seems that you have a high-level understanding on this. Then, what kind of strategies do you have to fine-tune the in-house information which is uploaded to the LLM model? Thank you for the good question. Whether it be machine learning or deep learning, having full data set does not guarantee performance. We need an appropriate amount of features to ensure quality. Then, what kind of data do we need, and what would be the proper amount of data? When we upload data to LLM model, I use just normal ways. I give the full model, and I enter more data or eliminate some of the data sets to see the change in its performance. I also look at the parameters of machine learning and do the fine-tuning. I am a clinician, so it could be a little bit of bias, but I enter data that I think is important. And sometimes when I write a paper, reviewers say, why did you exclude this data set when you uploaded the data to the LLM? So I would receive that kind of question from the judges of my paper. Anyhow, AI model selects important features, and I select important features from clinician's perspective as well. So we call this AI analysis, but we have to do a lot of human manual works. So in order to do the fine-tuning works, I upload or eliminate a lot of data sets from the LLM. I am Shin Jong-ho from Excel Therapy. I have a bit different perspective. When we choose LLM, what is your criteria? Because what is LLM? It already has a wide range of pre-trained data. And if you are going to upload in-house data and apply machine learning, the outcome of this activity could benefit you, or it could give you some negative outcome. So I think it is important to verify the LLM quality. You have to select the right LLM. Have you verified all the open source LLM? Do you have the comparison data among different types of LLMs? And if you get the outcome from LLM, can we use the data, or can we use the recommendation from LLM for prescription or treatment for patients? Because we cannot ensure that LLM always gives us a fact-based, accurate answer. Thank you for the question. There are many types of LLM model. It is, of course, important to compare different LLM models. As for open source models, there are a lot of research papers being published regarding LLM models. As for open source models, many of them are from a specific country. DeepSeek, Alibaba have also launched LLM models with good performance. In some of the papers, we can see some concerns over the use of LLM. So it is not easy to adopt an open source LLM right away. And models created by big companies like ChatGPT or Gemini and Claude, we had an issue related to the budget. I compared ChatGPT 5.4, 5.4 mini, and 5.5 mini. So comparing these different models of ChatGPT costed me a lot of money. If we compare all the LLMs in terms of their performance, we have to spend a large amount of money, and their performance does not differ much. So I'm not sure if it is worth spending that large amount of cost. So for now, we're using OpenAI model. We're comparing diverse types of ChatGPT models. This is what we have done. Please use your microphone. Do you have a separate process to utilize the recommendation from the LLM model for your clinical practices? The joint research team commercialized this model and sold this model to Gangnam Sacred Heart Hospital. That model is a bit different from mine. In the case of our model, the model I created is not highly likely to be used in my hospital because using EMR is a big issue. We cannot conduct some computation works through external server when it comes to EMR. So we have to establish a lot of systems and infrastructure in order to enable the connection with our in-house EMR. We have to do a lot of works. I heard that just drawing one feature into this model will cost us about 100 million Korean won per year. So if that's the amount of cost we have to bear, it's not really easy to use and apply our model in our hospital right away. This is why my first research was done in collaboration with a private company. Maybe we could apply this idea through a route like SCL, and once we receive recognition, we could try to receive the permission from the Korean FDA, for example. So thank you for many good questions, but please ask questions personally due to time constraint. Thank you.
To our great chair and wonderful speakers, we can finish session 2 strong once again. Thank you all of your contributions. Well, before moving on to the last segment, let's take a short break for 10 minutes. So we're going to resume 3:55. As you understand, the best has yet to come. So the best has been reserved for the last segment. So please stay with us until the last moment. We're gonna resume 3:55. 네, 여러분 잠시 10분만 쉬었다가요. 오늘의 마지막이죠. 세션 3. 3시 55분부터 다시 시작하도록 하겠습니다. 잠시 후에 다시 뵙도록 하겠습니다. 네, 여러분 장내로 입장하셔서 착석해 주시기 바랍니다. 저희 세 번째 세션 바로 이어가도록 하겠습니다. Segment of today right away, so please return to your seat and be seated. Well, once again, thank you for being here until the last moment of today. Well, we are very strict about intermission, but this time we skip intermission, so you're gonna listen to full presentation without any interruption. So welcome back again. I am very delighted to welcome you all to the fourth Infectious Disease Research Institute cooperation. Finally, we are about to begin the last session of session 3. The session 3 is about how we're going to develop the AI to develop the vaccine very quickly against the infectious disease. And this meaningful session will be navigated through by two esteemed scholars. So let me just take some time to introduce our great chairperson. First up, Do-gun Kim. He earned his all 3 degrees in biology from Korea University, and he's currently serving as the director of Center for Public Vaccine Development at Korea National Institute for Infectious Disease. And previously, he led the Division of Infectious Disease Vaccine Research at the same institution. Together with him, Professor Cha Ok-seok also will serve as our moderator for the last session. She earned her bachelor and the master degree in Seoul National University and doctoral degree in Chicago University in the Department of Chemistry. She's actually very busy these days. She's not only serving as the chair of the Science and Talent Subcommittee of National AI Strategy Committee, she's teaching at her alma mater, and also she's leading her own business called Gallux and the representative chair of Korea Science AI Forum. Over to you, Chair. 여러분, 큰 박수로 힘차게 응원.
Please welcome them with a big hands. Well, hello, good afternoon. So I'm the Kim Do-gun. I'm from the Korea National Institute of Infectious Disease. So this is the last session of both the Infectious Disease Research Institute cooperation. In this session, we are going to have a discussion about AI-based platform for rapid vaccine development against infectious disease. So AI-based development technologies will be introduced, and also the PPX and other platforms will be introduced. As MC introduced earlier, so without break, and we are going to continue for the presentations. And I'd like to ask speakers to make a presentation within 20-minute time frame, and that will be followed by 5-minute Q&A session. So please bear in your mind, please make a presentation in 20 minutes. So for the first and the third presentation will be supported by the Professor Seok, and the second and fourth presentation will be supported by me. So let me give a microphone.
Here is Dr. Paulina Brengel from CP, where she leads AI-driven data infrastructure for pandemic preparedness under the Disease X initiative. Her work focuses on integrating multi-domain biomedical data to accelerate vaccine development and enable rapid responses to emerging infectious diseases. Please join me in giving a warm welcome to Dr. Paulina Brengel.
Thank you. Great. Just checking you can hear me. Thank you very much for the introduction, and thank you very much for the invitation to speak here. It's been very interesting 2 days, and I hope this presentation will benefit and add some additional information. So CEPI was presented yesterday by my colleague Valentina, but just for those who were not here today, CEPI is a non-for-profit organization which is focusing on investing in the R&D of vaccine development for priority pathogens. And in my presentation, I will be speaking today about how can we achieve the 100-day mission for the pandemic preparedness through the pandemic preparedness engine in the Disease X. So how can you develop a vaccine for the unknown pathogen or for the Disease X? Within the Disease X program, within CEPI, we are operating and looking into viral families. We know that at the moment there are 25 viral families that can infect humans. In the meantime, if we were to develop a new vaccine, we also know that we need to go through all the stages of vaccine development, as you can see on the right. We need to be able to identify the pathogen through monitor. We need to discover and identify good genomic sequence of this pathogen, convert that for immunogen design, couple it with the vaccine platform, and then manufacture it, then take it through the clinical trial, safety and regulatory. So you can appreciate it's a quite long process, and the objective is to be able to do it within 100 days. So how do we do that within the Disease X program? We work in we're developing two products. The first product is essentially we're developing exemplar vaccines. So we are identifying a pipeline for vaccine development. First of all, if you look from left to right, the first step, we're prioritizing which viral families have a higher risk to spill over from animal to human. We take these viral families which are ranked the highest, and we will focus on those viral families to develop an exemplar vaccine. Then we're developing the immunogen sequence, looking into the structural biology, epitope focus, and we're selecting the best performing immunogens and feed them into preclinical studies, looking into expression, immunogenicity, and animal work. Then we would couple them with preclinical vaccine platform, whether it's an mRNA vaccine, adenovirus, or a protein, and that will really depend on the application of that vaccine, whether it's for response or for prevention. And then finally taking them all the way to clinical exemplars and clinical trials. So as you can see here, as we're down selecting the best performing and developing these exemplar vaccines, we're also generating a lot of knowledge. And we call this knowledge the R&D knowledge base for Disease X, or the vaccine library. I would like to focus on the knowledge base, on the second product, not the exemplar. How do we capture that knowledge? And this is where AI becomes relevant. And I like to refer to AI as a tool, not our goal. Essentially, it's like an electronic bike. I mean, it gives you a boost, but you still need to pedal. And this is how we're looking to utilize AI for the purpose of the R&D pipeline for the vaccine development. We're looking to use AI to enable the integration across multimodal data sources, essentially build this road, as you can see in this image, connecting between the monitor, the discover, and design on the different modules of the vaccine development. We're looking to use federated data sharing tools to be able to access data which is sensitive and can't be transferred from one place to another. And then finally, and most importantly, we would like to use AI for the speed, because that's how we're looking to achieve the 100-day mission. And just as an example, during the SARS-CoV-2 pandemic, Linear Design developed an immunogen for SARS-CoV-2 in 11 minutes, thanks to AI tools, something that would take days or sometimes even weeks if we were not using those tools. So this is why we're utilizing AI within our Disease X knowledge base. So essentially, we're taking the knowledge we're building on across these 25 viral families, we're combining it with the R&D knowledge we're developing, and we're putting it into an AI-powered engine, and we call it the Pandemic Preparedness Engine for Disease X, or PPX in short. And I'm going to explain a little bit how we're looking to train this engine and what sort of data we're going to feed it with. So as I've mentioned before, CEPI has a lot of partners across different modules and steps of the vaccine development. So whether it's monitor, looking into spillover, surveillance, and epidemiology information, discover, looking into the virology, phylogenetics, and escape models of pathogens, design, which looking into the biophysics, expression, and so on and so forth. And I'm not going to go over all of them. But overall, we have over 22 partners within the Disease X program developing knowledge. And in the bottom line, in blue, you can see some of these programs which are operating. And these are essentially the data sources which we're looking to integrate into the PPX. I'll give a few examples, and I will be mindful of time, so I might skip a few. But if we're looking into the monitor module, essentially, how do we decide which viral families we're going to focus on? We're working with University of California, Davis, which is developing the risk assessment protocol called VISTA. Essentially, they're collecting data of all viral families, and they're generating risk assessment across over 30 environmental risk factors of what's the likelihood for a virus to spill from animals to human. Then they provide us this ranking of viral families, as you can see on the right, which viral families are more likely compared to those who are less likely. As you can appreciate, it's quite a static list because the knowledge doesn't change very much. So we couple it together with Beacon, which is led by Boston University, which is looking and developing contextualized early warning systems. So essentially, the real-time epidemiology signals. And that's coupled together with this prioritization. And then it becomes a very strong tool of which pathogens to focus on to develop this knowledge. Another example, and I'll go through that extremely quickly, is on Discover. How do we identify the phylogenetics? So we're working with our partners within Argonne National Laboratory and University of Chicago on DXKB, which is the Disease X knowledge base, and also with Harvard University. And you can see on the right here, this is just an example of the program which is led by Harvard University, Professor Debbie Marks, looking into the likelihood of viruses to mutate and cause either higher transmissibility or immune evasion. And this will be the actual sequences we will be focusing in developing the medical countermeasures or the vaccine. And then, of course, the design, the immunogen design. I won't focus on that specific module because you have Jimmy Gallagher will be providing a full presentation, but this is essentially looking into how do we select and develop the actual sequence that will be used and incorporated within the vaccine, within the vaccines. So going back to the AI framework, what we're looking to do essentially is to implement agentic framework. And just very quickly, what is an agent? And I'm sure it has been discussed today. But essentially, it's a computational system that can interact with its environment, learn from it, from those interactions. So think about it as a smart lab assistant that not only can learn from the information that is generating, but it can also improve each time. So it's not just this kind of static, repetitive process. So what we're looking to do within PPX is develop this agentic framework where we will convert each of the modules of the vaccine development, whether it's monitor, discover, design, development, clinical trials, safety, and regulatory, into independent agents. And those agents will be interacting between them, but they will be covered in the orchestration layer with agents which are in red, which will be supervising. And essentially, this is inspired by the co-scientist agentic framework, which was developed by Google. And this is essentially what we're trying to establish. And that will be developed by Argonne National Laboratory together with University of Chicago. And essentially, like I said before, we're converting each of these vaccine development modules into independent agents. And I'll give you an example how we're envisaging for it to operate. So essentially, it becomes this collaborative researcher, and that can be used for vaccine development, for vaccine developers across different areas. So just as an example, using LLM, a researcher will be able to interact with the PPX and ask a question such as, I've identified this Nipah virus variant, please find the genome sequence attached. Can you please help me to develop vaccine targets? And then the agentic framework will be able to identify which of the modules or which of the agents needs to be activated. Is it a new pathogen? Is it a pathogen that already has immunogens and vaccines and just need to go through the clinical trial? And that will provide an output to the researcher. And just one thing to focus on, we're investing a lot of effort in the biosecurity layer of this engine because we're aware of the risks that if pathogens or models which can predict the mutation and cause a higher transmissibility leaks, we want to make sure that is well contained and they can't cause any harm. So the biosecurity is a very big part of the development of the PPX. Let's speak a little bit on the compute power. So essentially where this engine will be running. So while the bulk of the architecture is being developed and run on the Argonne National Laboratory in Chicago, we want to make sure that the engine is accessible across all the different regions in the world. So we're working with IDRIC, which is an AI factory in Germany, to make sure there is a regional access to the PPX. And also we're starting to work very closely with KISTI to make sure that also the Asian and the Korean potential exemplar has a local and regional access to compute power. Obviously, this is not equal across the different parts of the world, but we're trying to understand how can we make sure that there is still security, but also equitable access. So essentially, I will touch a little bit on the implementation plan. So as we're developing the global PPX models, which will be stored and set in Argonne, we're also developing regional exemplars. So as you can see here, just 3 examples. For example, the African exemplar, the European exemplar, or the Korean exemplar. And the reason for that is the following. Not only that we want to make sure that the actual computation is accessible locally, but we also want to make sure that the data remains locally. We're not looking to collect the data. We would like to be able for the data to remain locally, and only the aggregated form will be pushed and improve the models. It sounds perfect. Obviously, it's very complicated because there are quite a few challenges. So obviously, to establish trust and governance requires time, and this is something that we're investing quite a lot in terms of how do we make sure there is understanding about the data ownership and there is confidence in models to remain within the local region. And then finally, like I've mentioned in the previous slide, the other challenge is that the infrastructure is unequal. Not all parts of the world have the same access to the same level of supercompute and cloud solutions. And we're also working to understand how can we make sure that we can make that more uniform. And this is just one of the examples. So essentially, while we're developing the engine, we want to make sure it's actually usable. So on the 3rd and the 4th of March this year, earlier this month, we conducted a technical consultation to define the use cases of PPX in the African exemplar. We brought together a range of 3 types of stakeholders. First stakeholder is essentially pandemic preparedness and R&D vaccine development local initiatives. Some utilize AI, some don't utilize AI. The second one is basically supercompute and cloud solution companies which operate locally. And the third one is multilateral organization that can coordinate the development of such an engine. And we defined the PPX Africa exemplar use case, exploring how it can be used, who should be used by, and who will be coordinating the development of it. We assess the foundation for sustainable ecosystem, determine what is needed in the regional AI, data, and compute to ensure PPX can operate at scale. And then finally identified priority opportunities and gaps, and how can we potentially mitigate and address those gaps. So it was a consultation which it was very interesting to see the interaction between supercompute AI specialists and public health, because suddenly they understood the context between each other, as well as the public health organization that were able to understand how can they pull together the different groups. So just a few highlights. The consultation was co-led with WHO AFRO because we believe the implementation will be stronger if it's implemented by WHO regional office. We had some breakout sessions where we were trying to understand how can we achieve agentic framework using AI at scale. And then finally, we co-created a task force, and now we're going to start working together into developing it. And the reason I'm bringing this example is because essentially, we're looking to do similar consultations in other regions, because not every region has the same needs, and not every region will be using it exactly the same. We don't want to come with the solution and impose it. We want to develop it together with the stakeholders locally. So now it's my final slide, essentially, because the next thing that I'm usually being asked, okay, so when is this PPX going to be available? How can we use it? So a bit of time, bear with us. Essentially, in January this year, PPX was launched as a joint initiative between CEPI and the World Economic Forum, and it's being held by the Health Security Portfolio. And the reason I'm saying that, because essentially now we have 4 years to develop the engine, but we're not developing it in a vacuum, because the role that World Economic Forum takes here is essentially to make sure that this platform is implemented. So they will be responsible for setting up workshop series and roundtable discussions in terms of how can it be implemented and who should we work with to make sure it's useful. This is my last slide, just to summarize. So the way we're looking to respond to the 100-day mission within the DiseaseX program is by developing this secure managed access AI assistant for vaccine developers. We're thinking about it that it will become a global commons good as a virtual scientific collaborator to help researchers to advance vaccine development. And we're taking it very seriously for the implementation of security and access by design, and to make sure that the biosecurity guardrails are implemented so that there is no misuse of the data or the models that are being developed and utilized on this engine. And essentially, like I said before, I mean, we'll see the PPX that it will serve as a digital global commons for knowledge exchange for vaccine development and a key enabler to be able to achieve the 100-day mission for vaccine development. And that was my last slide, so over back to you. Thank you.
Thank you very much for the great talk. It is very impressive that you have this global federative system that can work together for like a future prepare this. Yeah. So we can take a few questions from the floor.
Thank you very much for a very interesting overview of the work. I'm curious in terms of you're building a AI-assisted workflow. What kind of trainings or what kind of support do you need to provide to the human researchers to enable them to interact with the AI better, knowing when to trust or when to be critical, so on and so forth? Just curious what kind of work has been done.
I think that's a very, very relevant question, and I think this is why we do see the human interface as one of the challenges. How do you trust the system? And that was one of the reasons we chose to team up with the World Economic Forum, because essentially they're looking to convene a lot of meetings to make sure and to provide this assurance in the system. It won't become straight away a ready-made assistant. We will have alpha and beta prototypes. And we're hoping through these different stages to bring on board more and more people, not only to test the performance, but also to be able to gain support and kind of spread the word of using it. So essentially to your question, teaming up with WEF, which has a massive reach to either pharma companies, governments and leaders, but also to make sure that we're actually taking our potential users through the whole process of the development. Thank you.
Let me ask you just one question. You compared AI to e-bike, right? So among these various steps you described, which one takes a lot of boosting, like a lot of efforts, human efforts, because the AI needs more improvement or so?
So the reason I gave that analogy, so I like cycling. I do a lot of road biking. So this was the reason. And I really feel like there is quite a bit of comparability with AI, because essentially it can make us faster, but we still need to make sure that whatever we're producing is verified. So the human in the loop is still very important, but it's for the expertise and not for the manual work. So essentially, I think it makes us faster. Hopefully, it will give us tools to connect things that will be very manual to connect it without those tools. But we still need to be able to identify the data sources. We need to be able to evaluate the data we're collecting, and we need to verify the output. So we still need to be connected to the whole process. So you still need to pedal, essentially.
Yeah.
But yeah.
So you need pedals in every step.
Exactly.
Right. Okay. Yeah. Thank you so much. Let's thank Dr. Brangle again for the great talk.
Thank you.
Thank you.
예, 그러면 두 번째.
Let us move on to the second presentation. The second speaker is from International Vaccine Institute. We would like to invite Dr. Byung-sik Shim, including IVI. He worked as script research and at the University of Georgia. He conducted research on developing vaccine for infectious diseases. Now he is developing next generation vaccine development to respond to new emergence of pandemics. Today, he's going to make presentation about SFTS mRNA vaccine development strategy. Thank you for the kind introduction. I am Byung-sik Shim from International Vaccine Institute. Today, I would like to talk about our strategy to develop SFTS mRNA vaccine for pandemic preparedness. My presentation consists of three parts. First, I will briefly give you an overview of SFTS virus. There's no approved vaccine for this, but at animal clinical trial level, we made various trials. There are many teams developing vaccines, and the relevant information is published. Based on the publication, I will share with you how SFTS vaccine is being developed. Thirdly, in September, we worked with IVI, and Professor Kim's teams to work on the CEPI Disease X Vaccine Library project. We were selected as the team to proceed with this project last September, so I will share with you the progress. As you know very well, SFTS is a tick-borne virus. It causes thrombocytopenia, and it causes high mortality up to 30%. But there's no approved vaccine for SFTS except for Japan. There's no therapeutics. In Japan, small molecular RDF injection has been approved as approved therapeutics for SFTS. This is a very important virus we have to deal with. This longicornis is the primary vector of SFTS virus. It infects not just humans but also various animals. It is caused by not just by tick bites, also through contact with the blood or body fluids of infected animals or patients. It first appeared in China and then it was reported in Japan, Thailand, Vietnam, and South Korea. Due to climate change, the habitats of ticks has expanded, so it is highly likely to become a pandemic. So we have to urgently develop vaccine for this. It has one phenotype, but it has 6 genotypes. In China, genotype FAD are dominant. In Korea and Japan, genotype B is dominant. And we have seen the emergence of F as well recently. SFTSV is an enveloped segmentalized negative stranded RNA virus. It consists of 3 structures like this. M gene, it consists of 2 parts, and we are coding this. Glycoproteins play a very important role. So this is one of the key antigens. GNGC glycoproteins are often utilized as key targets. Next, based on the published literature and papers, I would like to share with you the current status of the vaccine development, focusing on GN proteins. By using various platforms, researchers are developing vaccines. Many vaccine candidates are going through clinical trials and testings, including preclinical trials. I would like to share with you some of them. First, it uses VSV system as a platform. It creates our VSV platform based on the vaccine that expresses GN and GC, and it induces strong protective effect. And another interesting fact is that Cortland virus, another Vandari virus, can be targeted by this vaccine. And for challenge, it gives a very good level of protection. And it was confirmed that this vaccine has cross-protective activity. And when we vaccinated the Hanta GPA at SDS, there was no cross-activity. So in Hantan virus, the cross-protective activity was not found. So when we passively transfer this, we could see some level of protection. So GNGC plays a very important role. Next, this platform is based on adenovirus 5. It expresses GNGC and this platform was constructed. And when the expression was done in a form of single, it showed a good level of GN expression. After vaccination, interferon acceptor was utilized for GN group. 100 protection appeared, but for GC, no protection was induced. So this content is different from the previous slide. For GN, it induced the partially protective effect, so there are somewhat controversial aspect. Next one, use the DNA vaccine platform. This is a DNA vaccine platform that can express GNGC, and as you see in the middle, it gives us good protection effect. Interestingly, it targets non-envelope proteins. The T cell response was induced very well. So individually, they induced the cell responses very well. But it didn't induce neutralizing antibody. Interestingly, there was no GM protein here. They combined the envelope proteins, but still it showed good protection effect. And non-envelope proteins also showed partial protections. GNGC are important, but we cannot ignore the fact that the non-envelope proteins could also lead to some partial protection effect. Next one is subunit vaccine-based platform. This vaccine is known very well as inducing the immune response very well. At ferret module, when we challenged this, the protection was quite good. And WMC or leukopenia appeared a bit in the other types of platform, but in the subunit vaccine platform, there was no clinical signs of leukopenia or thrombocytopenia. And mRNA vaccine, they used the mRNA vaccine platform, and they combined the GN-head region, and they tracked the protective effect. Interestingly, the one that has GN-head showed good protection effect. And in terms of the weight loss, the one with GN-head showed much better performance. So the one that had GN-head only had good impact in terms of preventing weight loss. In this group, they used mRNA vaccine platform. They used a full-length glycoprotein. and it demonstrated long-term immunity. And in the case of challenge, it maintained the protection effect for quite a long time. I am sharing with you this information as an introduction to our platform development activities. When we challenge this with other types of virus, the cross-protection effect was induced. So we thought that by using mRNA vaccine platform, we could induce cross-protection effect. And similar trends can be observed in the other research activities, including the ones using machine learning model. Summary, so far, there's no approved SFTS vaccine, but by using various vaccine platforms, we have demonstrated protective efficacy. And GN glycoprotein is proven to be the most important target. Neutralizing antibody is, of course, important, but T cell responses are also very important for the complete protection. For SFTS vaccine, Pandavirus family or any other strains of families could be targeted as well because these platform-based vaccines are found to have cross-protection effect. Next, I would like to explain about CEPI Disease X vaccine library. We were selected as one of the institutes to proceed with this in September, so I will share with you what we do in this project. Due to COVID-19 pandemic, CEPI came up with this 100 days mission as explained by the previous speaker. So this mission is to deliver a safe and effective vaccine within 100 days of identifying a new pandemic pathogen. In order to become successful in this mission, we have to prepare a vaccine library. So in this picture, you can see a lot of the timelines. We have to establish the vaccines as soon as possible upon the emergence of a new pandemic. So we have different stages through which we have to secure the candidate vaccines. And if any pandemic takes place, we have to move quickly. And SFTS is one of the projects at CEPI. SFTS vaccine development projects are receiving support. We have 3 SFTS vaccine development projects. IVI, Pop Biotech, and these 3 institutes are working on this. As for Pop Biotech, they actually previously developed a COVID-19 vaccine-related platform, and they are going through preclinical. And for mRNA project, the timeline is about 4.5 years, and we have these 3 teams. We gathered together with the Seoul National University for a consortium and workshop last year. We discussed SFTS-related discussions for 2 days. We discussed epidemiology, animal model, and immunological assay harmonization. So it was such a meaningful time. Let me share with you the project information. The title is Development of an SFTSV mRNA Vaccine as an Exemplar for Virus Vaccine. So it targets SFTS as a family of virus vaccine. This is the budget for the period 4.5 years. According to the 100-day mission of CEPI, we will establish a mRNA vaccine platform to quickly respond to the future situation. So the goal is to complete the efficacy assessment while advancing this project to phase 1 and 2 clinical trials. Dr. Man Gi-song is the principal investigator, and I am part of the technical leaders. These are our partners. We have these 4 teams in the consortium: IVI, KDCA, Seoul National University, and ST Farm. They have their own strengths and they have powerful tools as well. Let me explain about what they do. IVI, they successfully commercialized 2 vaccines. Now, what they developed are being used very actively for vaccinations. They have abundant experience in vaccination. It leads the project and it is involved in the preclinical immunogenicity efficacy evaluation as well. And KDCA team is helping us a lot in this project. First, KDCA is working on SFTS virus antigen design and optimization. It is also evaluating the vaccine-induced immunogenicity. Sarah-validated ferret model is very important. So KDCA is going to provide us with strong support for using the validated ferret model. Next is Seoul National University. We have 2 teams by Professor Kim and Professor Tang. They have linear design platform, UTR design platform. Before optimization, RNA structure was different. After optimization, RNA structure has the form of helix. Now, secondary structure of mRNA is also being considered in this project. At Seoul National University, they published a paper and they have a patent about this. mRNA polytail decreases, so they can enhance the RNA stability, and they screened about 200,000 elements here. And ST Farm, they have SmartCap and ST-LNP technologies. And ST Farm has GMP manufacturing system. So at clinical trial stage, GMP will be done by ST Farm. And they have experience with going through clinical trials with other similar vaccine-related projects before. So to summarize, we have mRNA vaccine platform, and ST Farm has SmartCap, and Seoul National University has patent in this area as well. And as an interesting gene, we are using SFTSV full glioproteins. We have 2 strategies. First, KDCA will change the amino acid to enhance the stability of the structure, and Aminex optimization is going to be done. So the optimized sequence will be sent to Seoul National University, and then Seoul National University will optimize the structure and codon of the mRNA vaccine platform. Our scope ranges from the design of antigen and preclinical and phase 1-2 clinical trials. It includes toxicity assessments and other phase 1 and 2 clinical studies. This slide shows the strategy of our consortium for antigen design and selection. It has been about 6 months since we began this project. We are nowadays optimizing RNA. We selected backbone And through amino acid mutation,
안정성이 올라간 그런 표현들을 확인할
we
수가
have
있습니다.
more
그래서
stabilized sequence.
세계에서 내게 된 top priority 시퀀스를 지금 선정하고
And 3 to 4 top priority sequences are currently being selected. And we are optimizing mRNA.
추가 탄력 실험을 하고
After that, we will do some more selections.
SLP를 현혹도가 그 포뮬레이션을 같이 해서
And we will do the formulation of SDLMP before finally selecting the candidate.
세 번째 백번 셀렉션을 하기 위한 결과고요.
This is the outcome we used for backbone
이렇게 바이러스가
selection.
GPA 익스프레션
For
레벨이
same
달라요.
virus, they have different GP level.
알파 시퀀스의 GP 단백질의 발현 레벨을 테스트
So we tested the level of genotypes expressed in the sequence.
있기 때문에 저희가 좀 높은 발현율이 높은 걸 좀 선정하고 싶었고
So we wanted to select the one with higher level of expression.
이 질병 관련성에서 genotype B에 대한 B에 대한 GP 시퀀스를 다 증명해서
The GP sequence was also provided from KDCA, and we looked for any null types, and we found one null type. This is from a human isolate. This is the strain we have been using. We checked the expression and found that B1 showed better expression, so we selected B1. They are both consensus sequences, but at B1, there was difference in amino acid. So among genotypes, there was a difference. And based on B1, Seoul National University worked on optimization. It was a linear design strategy. Professor Chang from Seoul National University developed a VaxLab program. It shows secondary structure, but also codon usage and C' optimization. It is a comprehensive platform. By using this platform, we created 3 optimized genes. We combined them and made a comparison for number 2 and 3. The expression was induced very well. By using this powerful tool, we were able to increase the level of expression. And by using B1 backbone, we applied protein structure modeling, and we found that the structure was more stabilized. So we didn't touch on the area that was supposed to the surface, and we conducted amino acid mutations between GN and GC, and we consider the stability and interface energetics and solubility as well. Amino acid was mutated compared to Y type. We have not yet found a high expression. But number 10 showed the level of expression similar to the previous slide. It is a different story from inducing neutralizing antibody. We are conducting additional study and research. We will secure 3 to 4 sequences more, and we will use VaxLab program to secure from 20 to 60 candidates and conduct another test before selecting the final candidate. And summary. So we need to develop vaccines for SFTS. We don't have approved candidates, so we will make preparations very well so that we can successfully go through phase 1 and 2 clinical trials. We have high expectation. This is all for my presentation. Thank you for your attention. Thank you for your presentation. Due to time constraints, I don't think we can receive a lot of questions. We could take some short questions from the floor. So I'm interested a lot in this project. You said GP expression was done and detection was done through GC. Yeah, both GC and GL. You did GN and GC together, but for detection you used GC. For GN we had some issue, but we found the expression of GN. So for GN, the protection was good, but for GC, there was a data showing worse outcome. So I was a bit concerned about this. Another thing is related to optimization. In the Western blot, you said optimization was done well, but I don't think it was really truly optimized. The white type seems to have high efficacy. What do you think of this? For white type gene, when we mutated it with the secondary structure, although for the same white type, the optimized expression was very good. Yeah, but in my opinion, I don't think the optimization was done very well. But if you look at different timelines, we can get better results. Thank you for your comments. Dr. Jung has a question. Thank you for your presentation. Let me ask you a simple question. mRNA vaccine itself has high level of expression, and in 3' mutr, you have viral neuron risks. And what is the reason? So being able to increase expression means that we can use a smaller amount of vaccine, which enhances safety. And the higher the expression, the higher level of neutralizing antibody impact. You might have more questions, but due to time constraint, we will move on to the next presentation.
감사합니다.
So our next speaker is Dr. Jimmy Golliher. He's the Chief of Translational Science at Houston Methodist Research Institute. He's a leader in AI-driven immunogen design and synthetic biology. Okay. His work integrates machine learning, directed evolution, and high-throughput experimental systems to develop scalable platforms for antibody discovery, vaccine design, and protein therapeutics. Today, he's going to tell us about pandemic preparedness, vaccine design, and antigen engineering. Please join me in welcoming Dr. Jimmy Golliher.
Thank you. Thank you very much. Wow, what a symposium. I really appreciate the hospitality. This has been amazing, and the food here has been incredible. So if you're not from Houston, you probably have never heard of Houston Methodist. So I thought I would give you a brief overview of where I'm from. So Houston Methodist is a hospital system that is only found within the city of Houston. We have a flagship hospital at Texas Medical Center, and then the Research Institute that you see here. We have 8 full hospitals around the community, along with 120 clinics. We are a nonprofit hospital, and we do a lot of clinical research. So we've done about 523 clinical trials, 177 of those are investigator initiated. We've got a number of different types of therapeutics in the pipeline, and I think what sets us apart from a lot of academic or healthcare institutions is the fact that we can manufacture GMP on site. So we can really go from bench to bedside without leaving the house. The Research Institute is currently this 400,000 square foot building. We're in the process of planning out another 700,000 square foot addition that we should be going to soon. And we've been rated the #1 hospital in Texas for 14 years in a row now this year. We have a lot of clinical translation infrastructure or translational infrastructure, a lot of imaging. On the first floor, my protein engineering lab makes up about half of the 6th floor. We have small and large animal vivariums on the 4th and 5th floors, and we can go to BSL 3. The 12th floor is where most of our GMP suites are, with the exception of our Johnson Cell Therapy Center, which is over in OPC, over here in red, and there we made 46 CAR Ts last year. So translating discoveries is a part of our DNA. Today, I'm going to show you the same slide that you've seen 3 times in the last half an hour. The concept of vaccine libraries is something we've been working on with CEPI for a few years now. And so while Paulina didn't get into the exemplar part of this, I'll briefly go over that. So within each of these families that are known to infect humans, we look to be able to engineer vaccines for a particular virus for which there are tools for. Because if we can't engineer vaccines against those targets, we have no chance in the world of engineering a vaccine to a disease X. And so we really go pretty deep on an exemplar vaccine, and then see how far we can push those mutations to other members of the family while learning as much as possible. Currently on the hook for 6 of these viral families, and we are building 10 to 15 vaccine candidates within each family. To do this, we use something that we call the Vaccine Immunogen Prediction Neural Network for Disease X, or just VIPNetX. And during Paulina's talk, she talked about what we do made up a single bubble, the antigen design. If you blow that up, it's actually a collection of many different models and things. We don't believe that a general model is generally good, so we build different models that specialize in very different tasks that allow us to engineer immunogens very, very rapidly. So the first part of this is, it starts out with understanding what your target antigen is, pre-processing it, and being able to annotate it and know what you're working with. Then we have sequence-based models that tell us more about the phylogeny, how related it is to something. We look at structure and stability, so 3-dimensional pre-fusion stabilization of vaccines, and then what do B cells and T cells epitopes look like? How can we make sure that we're preserving those? And then we put this all together in an antigen design model. While all of these pieces exist, there is a lot of human in the loop, as Paulina mentioned. A lot of this is manually moving things from one place to the other. So with the advent of the pandemic preparedness engine, we hope to have agentic control over these tools so that we're able to use them very rapidly and without humans as much in the loop. Our immunogen design workflow is shown here. The only thing on this slide that we're not really responsible for is target acquisition. That comes from CEPI or other partners. Once we have a target, then we need to be able to annotate, extract features, design immunogens for B cell and T cell responses, and then go through this multimodal validation and refinement stage where we're actually taking the predictions from silicon and putting them into carbon and actually testing them. And so that can be cryo-EM, that can be animal protection, can be any number of assays that are happening in the real world. Once we are satisfied with a candidate or a number of candidates that have gone through BSL-4 or BSL-3 challenge, we then transfer those over to developers. And in the case of the CEPI project, that is pre-identified CEPI partners. We've in fact worked with folks here in Korea already. And on the bottom here, I'm just showing our consortium. This is not just Houston Methodist. We work with UTMB for BSL-4, Jason McClellan at UT Austin, the La Jolla Institute for Immunology for T-cell work. You all probably know Alex Setti and Alba Grafoni, the University of Chicago, who Paulina mentioned, JCVI, as well as Leipzig. Now, we think that the next generation of mRNA vaccines will likely have B-cell and T-cell components. And so to get a B-cell response, you need something to get outside of the cell, more likely than not. And so we really take advantage of being able to engineer immunogens to be secreted or be on the cell surface. Now, of course, you're also still going to mount a T-cell response to those. On the other hand, we think a lot of vaccine durability may come from immunodominant protective epitopes that are in maybe non-structural proteins, and so we've developed strategies to be able to engineer those, or pieces of those, which I'll talk about later, and shunt them through the proteasome to hit MHC class 1s. We can also go through the lysosome for class 2s. So starting with how to design an antigen. From a protein engineering perspective, it's pretty simple. But to teach a computer how to do the same thing, you have to sort of put things into bite-sized chunks. And so the first thing we do is classify the virus. Where does it belong? What is it related to? Do we know anything about that family? Then we need to be able to annotate the genomes and understand what is the fusion machinery, what is the attachment machinery, what are the non-structural proteins, and then extract features so that we know what we can engineer, when we can engineer it, and what to just leave alone. From that, we then prepare three-dimensional models and perform stability mutations in silico there, or a variety of other tools. To annotate a protein, you might say, well, you just go to Uniprot and download the annotations, but in a disease X scenario, you're not going to have an annotated genome. There are a number of tools that can annotate parts of proteins, right? Signal P, you've heard of dozens of these. However, they're all written in different languages. They're very difficult to get to work together. Their inputs, their outputs are all very different. We have actually put those into a pipeline and have it containerized and it works. But we wanted to ask the question, can we train a protein language model to be able to do this almost instantaneously? And so I had a graduate student take ESM2, which you've all heard of, stick a classification head on it and a hidden Markov model, consolidation model, and be able to predict 10 different types of protein annotations. And through this, we're able to do about 2,000 proteins in less than 3 minutes. That then becomes the pre-processing step for us to begin our hardcore stabilization or engineering to go through the secretory pathway, through the proteasome, the lysosome, whatever the case may be. So the performance on this model is state-of-the-art in 6 of the different classification tasks and is competing with the others. So this was a nice first project for one of my graduate students, and it is something that we are now putting in the knowledge base that Paulina talked about, and will be a tool that is used moving forward. Here's an example of how this is used. So this is everyone's vaccine or favorite protein to talk about during vaccines, SARS-CoV-2. This was not in the training set, and you can see that the model pulls out almost everything very faithfully, disordered regions being the exception, and those aren't necessarily important for what we're doing. We also want to know where do antibodies bind? And so looking at SABDAB or our own internal databases, we want a way to query where antibodies bind certain immunogens, because we're going to use antibodies as conformational probes when we go to engineer these in the exemplars. And so we've developed a dashboard that allows us to visualize and then select monoclonal antibodies that we'll then use later to ensure that the engineering that we're doing isn't disrupting epitopes or the conformation of the antigen. We also need to know what the real surface is. And so there are solvent accessibility surface area calculators out there. The issue with them is that they take probes that are just randomly generated in space, and viral glycoproteins have giant voids in them because they're metastable and adopt different conformations. And so the standardized way of looking at what the surface is puts residues in the middle of the protein, marks them as being on the surface, and so unengineerable. Whereas a protein engineer, we could just go in there and pick it, but remember, we're trying to teach a computer how to engineer a protein, and so we need to make sure that it's got an accurate representation of where to put mutations. So we can annotate, we know where antibodies bind, we know what the surface is. We obviously don't want to mutate residues on the surface of a viral glycoprotein because that will create or destroy protective epitopes. We don't want to create an epitope that doesn't exist in nature and raise an antibody to something that isn't going to protect anyone. And so once we have all of that information, we then, on the B cell side of things, want to pre-fusion stabilize the attachment or fusion machinery. And so this is an example from Jason McClellan many, many years ago, pre-fusion stabilizing RSV, and I believe someone in here did the NHP work for this study. But we want to pre-fusion stabilize the glycoprotein so that we hold it in this conformation and elicit antibodies that will bind that and keep it from being able to either bind the receptor or fuse with the host cell. One of the ways that we do this is with a convolutional neural network that Raghav in my lab built about 8 years ago when we were all in Austin. And what it does is basically takes an amino acid and deletes it, looks at the surrounding environment, and says what amino acid makes the most sense here. That was great for a first pass, but then we found that we could train this model on very different types of mutations. So we started with a single antigen design circle, and then I'm showing you another set of 5 algorithms inside of that. So we have an algorithm that predicts just disulfide bonds, just salt bridges, how to fill a cavity, all of the things that an immunogen designer would do rationally. The 3D convolutional neural network does that specifically. Once we once we have a prediction, then we need to get it into carbon and test it out. And so 2 of the ways that we do that are through what we call native display, where we take the full-length glycoprotein, we express it in human cells, and then we use monoclonal antibodies to monitor the expression and antigenicity of a particular variant. The other thing we do is tether ectodomains. So old school immunogen design would take and make soluble ectodomains and engineer them one at a time, like how Jason would put disulfides and DS-Cav, put cavity filling mutations. We make it high throughput by tethering that same construct on the surface and embedding it in the membrane, and that allows us to then use things like high throughput flow cytometry or FACS, cell sorting, to be able to probe the function of these mutations. This is an example of Lassa fever virus. Just really quickly, we took our version 1.0 tools, we built 300 variants, and we found a number in blue where we were over 2 times expression or pre-fusion stability over wild type. Once we had those, since Lhasa has a lot of antibodies. We then used a panel of 16 antibodies hitting different regions of the GPC and asked which of these combinations is better for expression, prefusion stability, and just overall stability. And we found a single variant that had 4 prolines in it. And then Jason solved the structure of this. In blue, you see the soluble ectodomain. The antibody 12-1F is here in orange or yellow. And this was the first, or the highest resolution structure of a Lhasa GPC, just under 3 angstroms. But importantly, it doesn't have a non-native disulfide bond, which all of the other ones that had been engineered to date contained. We then put several of these in animals, and what we found is that all of our immunogens were 100% protective. You see a couple that die here. They never had viremia, they never had weight loss, they just didn't wake up after being anesthetized. That sort of takes you through the version 1.0 of the platform. We use a convolutional neural network, make some predictions, screen those individually against a couple of monoclonals, then use a panel of monoclonals to probe the structure in high throughput. And in 8 months or so, we were able to do this to 8 other viral glycoproteins. We started here with Lhasa. This took us about 3 months to get through. Hunin took us about 3 weeks. H5N1, HA, and NA, both about 4 weeks total. And RSV, not that long, Nipah, and chikungunya. So these tools were great. We were able to get to immunogens very, very rapidly, but we thought, what are we missing? What are our models bad at, and how do we make them better? And so we came up with the concept of doing DMS on these, deep mutational scanning, where we would take unbiased scans, take those data, feed them into model, create new test sets, and have refined models out the end. We chose only to hit residues that were in the interior of the protein. Again, we didn't want to create or destroy important epitopes. And for the first pass at this, we did proline scans and size scans, whereby we would take an amino acid and either make it smaller or bigger, or change the chemistry. That made the number of mutations that we could make in a protein a bit more manageable. We have since moved on to doing full DMS minus cysteine. The way this goes is we will take and order DNA. The only thing we don't do in the laboratory is synthesize DNA ourselves. We'll then assemble those with an Echo 525, an acoustic liquid handling robot. We'll then inoculate in 384 well plates, sequence on Oxford nanopore, and then pick the good clones. A single run by a single technician usually yields about 720 unique variants, and we can do about 9,200 of these a week. An example of doing this is with SFTS, which you just heard about, so I won't go into the detail there. The surface of this protein, or residues that we are not going to touch, are shown in gray. And this is now a 3-dimensional heat map showing every mutation that's not on the surface, the highest expression or prefusion stability at each position, and we can start to see mutational hotspots where the protein likes to be mutated, and we can see places where it definitely does not like to be mutated. So we can use these data to fine-tune models. This is just another protein language model that uses structure tokens. And when we take the data that we generate in the laboratory, we can then use those to predict prefusion stabilizing mutations as well as expression mutations, and we have state-of-the-art performance for both of these. I'm going to move through this very quickly because I'm about out of time. We do something very similar for T cells, where we look for T cell epitopes, conserve T cell epitope regions, put those into mRNA, we test them, and rearrange them in silico in different ways and try to come up with more stable ways of expressing them. and that sort of puts things together, a B cell side, a T cell side. This is my CEPI consortium. I mentioned them earlier. This is part of my group. We're 43 people strong at Houston Methodist. This is our funding, and I'll take any questions that you have. Thank you.
Thank you very much for the wonderful talk. So we can take like one question from the floor.
Well, thank you for the wonderful presentation. So actually, Phylum Map version 1.0, that is the open source. And I believe that is not open source, right? That is not openly available in public.
Well, the original convolutional neural network is the individual components that have been fine-tuned to the different types of mutations has not. We're in the process of publishing that now.
So in the future, what is your plan? So could you share your future plan?
Yeah. So I think in the future, what we would like to do is create the antigen design model that is used by CEPI's PPX program and open that up to users within the CEPI network to be able to engineer vaccines.
Okay. So it's very impressive that you have this whole toolbox in Houston Methodist, and I was very impressed by the work on SARS-CoV-2 spike protein. You were trying to stabilize pre-fusion form compared to post-fusion form by AI, and then validating in the surface display. And I actually, one of my colleagues at SNU is developing a Rosetta MPNN variant that can stabilize one form compared to the another form, and she has like prediction data, and I thought that it would be really great if she can compare your experimental data with their predictive data.
Yeah, that would be great. We would love to. We actually invented this platform during SARS-CoV-2, not for pre-fusion stabilization, but we were wanting to know whether or not the monoclonal antibodies that we were developing were being escaped by the variants of concern in real time. And so in those experiments, we were really, whatever our sequencing out, we sequenced 300,000 COVID genomes, right? And we would take a look at mutations that were starting to pop up, throw them into this platform and say, okay, these are escaping this class of antibodies, these are escaping these. Maybe we don't need to spend millions of dollars trying to develop an antibody that is going to be irrelevant in 6 weeks.
Yeah, and I think that's a very valuable data because we can feed those data back into the AI algorithm to improve.
Yeah,
right?
Absolutely.
Yeah. Okay, so otherwise, let's thank Dr. Jimmy Collier again for his great talk.
Then we will invite the last speaker, Professor Tae-sik Chung from Sangmyeong University. Professor Chung is a professor at the Software Convergence Department at Sangmyeong University. He is also the CEO of InSilicox. He is researching data integration and prediction model development. These days, he is researching development of animal alternative AI models based on human organoid and animal model data. Today, he will share with us his experience regarding the animal alternative AI models. Nice to meet you. I am Tae-sik Chung from Sangmyeong University. I am also the CEO of InSilico. It's impressive that we have many audience still staying with us at this stage as we are approaching the end of the session. First, regarding the limits we experience in animal experiments, you would know very well. So, through animal experiments, we conduct a lot of trials, but 90% of animal tested drugs fail in human clinical trials. Considering international conventions and considering the recent trends in regulatory authorities, we recognize very well that many of the animal tested drugs have failed in human clinical trials, and we're working on alternative ways to conduct the trials. And the NAMS concept has developed and has been advanced very well. It means that instead of animals, we need to conduct the test by using human relevant biological data. It reflects the global trends and needs, and we need to expand this concept to move towards closer towards the human relevance. Under NAM's concept, we can culture human organoid, a 3D organoid. As you know very well, lung, intestinal organs, and the liver can be created as human organoids, and they reflect human physiological characteristics. It can be a good alternative to animal testing, but there's a big issue. As you know, data from organoid is not seamlessly connected to preclinical or clinical data. For decades of period of time, there has been efforts to integrate organoid data with preclinical and clinical data, but it has been very difficult. So how can we analyze and integrate the tens of years of organoid data with preclinical or clinical data? So we need to land these data sets in the AI model. That includes animal testing data, organoid data, as well as clinical data from hospitals. All of these data sets exist without standardized format. However hard we try to enter these different data sets into AI model, it doesn't work very well. And although we can use AI to make prediction, it's very difficult to use these varieties of data sets. To solve this issue, the Ministry of Health and Welfare launched a KAI project. In Silicos is the co-research institute along with Korea Research Institute of Bioscience and Biotechnology. The project title is Building the Efficacy and Safety Evaluation Platform based on Organoid Digital Twin Integrated AI Model. Especially organoid data and preclinical and clinical data should be integrated so that we can evaluate or predict efficacy and safety of drugs without animal tests. This is the key objective. To do this, we are working with Severance Hospital for clinical data, and we're working with Korea Institute of Chemistry Research for organoid data. And we are working with Korea Research Institute of Bioscience and Biotechnology for preclinical data. So in this way, we launched our efforts to create AI model as an alternative to animals. Then how does this model work? You might have questions. The most important keyword is federation model. This is based on PIN model. We entered the animal test models and organoid data to secure enough amount of factors which pass through the PIN model, and we were able to predict efficacy and toxicity from this prediction model. Let's look into details. PIN model itself gives us information about equation of physical data, including pharmacokinetics. The pharmacokinetics or toxicity information are part of the in-house information. Such information is applied to PINN model. And we fine-tune the external information and in-house information. It shows the equation. So actually, the PINN is applied with the pharmacokinetic ordinary differential equations. It gives us more robust accuracy. Compared to conventional AI model, there are many conventional AI models, but KAI projects platforms based on PINN model could solve a lot of issues we experienced in the conventional AI models. So we can use both in-house information and external information to extract more explainable data sets. And intuitively speaking, ranging from small animals to humans and primates, we can integrate all the organoid data. So standardization is very important. As mentioned before, different data sets are not standardized. So our important aim is to build standardized data sets and develop AI model. So data types include whole exome sequencing, RNA sequence, and all these data sets are standardized, and we collect all the data sets, including clinical phenotypes, and we refine the data sets. Then we can get toxicity prediction, efficacy prediction, as well as pharmacokinetics and pharmacodynamics modeling. So how do we express this concept? At KAI project, we say that we will establish a pipeline for data standardization and quality assurance. So the standardization refers to having a common data sets. Different sources of data can be combined with a common data model for standardization. So our goal is to achieve data standardization at a level to be able to submit the data to the regulatory authorities. So common data model, this is the concept we are applying to this project. Fourthly, when it comes to AI model, they have to go through verification process. The prediction based on organoid is sometimes not consistent with the clinical or preclinical data outcome. Actually, there should be correlation between organoid models and preclinical or clinical experimental data. So also we have time sequence data. So we have to continuously validate whether there's correlation between the organoid models and the historical preclinical and clinical experimental data. This is an example ranging from small animals to human primates. We have a lot of image-based data sets like this, but as for preclinical data, they consist of numbers and texts. So we have to enter various types of data sets into the model and produce good prediction. Then how can we train this model with different data sets and draw out a good quality prediction? In the case of image, organoid image, there are many conventional AI models. Through those models, we can extract features, and IS50 or AOC and other numeric data can go through neural networks, and then we can get the features. And we have the embedding fuse layer. Going through the fuse layer, we can get image, we can train the model with image data and time series data along with numeric data. So the different data sets can be combined. And then, so far I talked about the model-based efforts. So advancing the model with in-house data. Then we could enable agentic AI service to monitor the situation, and the administrator can track the content through a dashboard. So the next step is to build a pipeline for agentic AI. So building, combining, standardizing, and monitoring the data can be done through agentic AI. We could use external LLM API, or we could use our own foundation model and fine-tune the layer with in-house information. But we need all these efforts. So we need to utilize the models, and based on AI Studio, we should build an agentic AI. External information is very important, and in-house information is of course important. At KAI project, we will utilize agentic AI in this effort. Through this project, we can achieve three things. Firstly, regarding the trustworthiness of AI model, we can identify how trustworthy this AI model would be. And if we succeed in this project, we will be able to set a screening strategy to reduce animal testing. And if all this is done through agentic AI, then all practical elements demanded under NAM's concept will be achieved through this agentic AI model. And there are various scenarios, not just bio industry, but robotics, physical AI, all other many industries will be able to use the system. And both in-house and external information, including preclinical and clinical information, could be entered into the system. And with the help of LLM and machine learning model, we could produce better prediction for the drug efficacy and evaluation of toxicity. So the testings and trials and could be done more efficiently in the future. So, so we're working with KPAC and digital preclinical platforms. We have internal AI agent and external AI agentic elements will be combined. Then what about internal LLM? How do we establish internal LLM? We don't have a supercomputer. We will use the existing foundational model and fine-tune the model with our in-house information. We will be able to fine-tune the model at a level to provide the relevant service. We can do the verification at GPU level. KPAP and InSilicox and academic institutes signed an MOU. At MPU level, we could fine-tune the elements specializing in bio-LLM. So in the era of, so we will come closer to the bioagentic, the era of bioagentic era. So it is important to establish a system to verify this platform. This is a prototype. Within KPAC, we have digital preclinical platform. Models used in this platform is based on federated learning model. We use CNN for image. For time series, we use neural networks. And AI assistant here is from AWS Bedrock or ChatGPT. It's not the external model, but the AI system here is based on our foundational model. And we are establishing this platform. And diverse fields of expertise could use this kind of agentic AI platform. We could use RAG methodologies. This platform shows elements related only to preclinical, but this is not confined only to preclinical areas. We could apply this platform for responding to infectious diseases as well. We have to be able to use information both from outside and from in-house information. We could use RH methodology to reduce hallucination. And the internal prompts can be used as the training data sets for the model. So continuous learning and training of the model is very important. So this is one of the areas we're focusing on. You might have some questions about agentic AI. So I'm sharing with you this slide. The external LLM model was compared with our in-house fine-tuned LLM model. We could connect them through a pipeline, and the researcher can set an assumption, conduct a trial, and verify the results. We have our in-house solution, and we are working with IDRIC. We have completed fine-tuning of this platform. And this is not something we can provide as an external API. We can establish this platform as an in-house platform, and researchers can configure the setting, and they will be able to make a good use of this system. The most important part here is the validation. We cannot rely 100% on this AI platform. However, in your research and trials, AI will make it easier for you to make better prediction and validation of your research activities. Here, of course, you can pull various articles or paper-related information. Currently, I am studying natural resources bank and natural resource related areas. So I ask here to the AI system, do you think what kind of positive impact a certain plant can give to so whatever question you enter here, that you can get answer from the platform, just like when you use ChatGPT or Gemini. It is like using ChatGPT or Gemini-like functions as an intranet feature. And the prompts need to go through human validation. Wrong prompts or wrong information should be corrected. And the system will go through the training continuously. Thank you for your attention. Thank you for your presentation. Then we could take question from the floor or comments. Thank you for the presentation. I developed an alternative method when we produced toxicity data. Interlaboratory validation is very important. After making the prediction model, we need to go through verification. Do you plan to verify the model? And what do you think about the verification process? And Korea, and you said that you can produce data at a level that can be submitted to Korea FDA. How does Korean FDA think about your idea, creating a data through the AI system at a level that can be submitted to the regulatory authorities. Yesterday, I made a presentation about our project. We conducted a retrospective and prospective cohort study related to this one. It is about toxicity model. There were limits in what we can do by using AI model. By using actually for that AI model, we have a system of cross-verification between the outcome from AI model and the actual clinical toxicity related data. So we of course have a plan for verification by comparing the outcome of the AI model with the actual clinical data. And regarding the level or quality of data, what I meant was that we want to enable the platform to assist you in preparing information or data at a level that could help you a lot in submitting your documents to the regulatory authorities. Thank you for the presentation. I am Kim Jae-wook from International Vaccine Institute. Through organoid model, we can measure some values related to toxicity. As for vaccine, when do you think it will be possible for us to measure various values related to vaccine? For now, we are working on mRNA platform project with other academic institute, but that project has not yet fully begun. The data includes time series values related to sequencing or expression. If such data is used for training this model, then we will be able to measure some values, but this is going to be based on regression. My major is computer science. I don't know what this actually means from biological perspectives, but if you give us the data, for example, if you give us the feature about expression, we could give you some points about what kind of outcomes the machine learning model created, but this is all I can say. Maybe I could give you more detailed answers later on. Do you have other questions? Then in the last session, we learned about how AI is used for rapidly developing vaccines for infectious diseases. Thank you for your presentation, and we will conclude the session now. Thank you very much.
Well, thank you. Special thanks goes to not only the speakers but also the co-chairs. So please give them another big hand, please. 네, 정말 수고 많으셨습니다. 감사합니다. With this, we finally bring an end to two-day-long journey. And to bring an official closure, I'd like to invite once again Director General Young Gi-jung of National Institute of Infectious Disease for his closing speech. Please welcome him with a big hand. 여러분, 정영기 소장님 모시고 폐회사 청해 듣겠습니다. 박수 부탁드립니다.
So this is the last the program of this international symposium. Still, we have a high number of attendants. And as an organizer of this event, I'd like to express my appreciation to every one of you. So we have discussions about AI the based approaches. So I could see the great interest of participants about AI, and I am motivated to provide better programs with better topics next time. So we had discussions about the next generation R&D strategies for AI-driven antiviral drug developments. And regarding that, we have constructive discussions. And all the suggestions and ideas presented in this symposium will be reflected in our the future policies and future efforts so that we can fasten our effort in order to develop vaccines for the disease X in the future. I'd like to extend my sincere thanks to all the speakers, and my thanks also go to the moderators. And in particular, my special appreciation goes to all the participants. Thanks to your active participation, we could have a successful hosting of this international event. So, well, in the closing, I'd like to express my sincere thanks to the Egan He and the Foundation and the organizing organizations for wonderfully preparing this event. With this, I declare the conclusion of the fourth Infectious Disease Research Institute Corporation. Thank you.
Thank you. Thank you very much. As he mentioned, your attention and your contribution make our two-day-long symposium a great, successful, and truly meaningful. Thank you, everyone, once again. And we're going to put more efforts when we come up with the next year's event. So we sincerely wish to see you next year. Thank you, everyone. Safe journey back. Thank you. 여러분, 대단히 감사합니다. 내년에 뵙겠습니다. 안녕히 가십시오.